Assalamu alaikum and good evening. ICAZ-2021, PhD scholar from Government College University, Faisalabad. Welcome you all in the 4th International Conference on Applied Zoology 2021 on the behalf of Organizing Committee of the Applied Zoological Society of Pakistan in collaboration with Quaid-e-Azam University, Islamabad. It's a pleasure for me to be the moderator of 7th session of the 4th International Conference on Applied Zoology 2021. Our session contains the talks from the fields of drug delivery, neurosciences, cell biology, and nanotechnology. Let's start our session with our first presenter, Dr. Fozia Anjum from Government College University, Faisalabad. She is going to present her work with title Date palm mucilage encapsulated with silver nanoparticles for colonic drug delivery. Please, Dr. Fozia Anjum.
Bismillahirrahmanirrahim. Here is Dr. Fozia Anjum presenting some research work which is entitled as Date palm mucilage encapsulated with silver nanoparticles for colonic drug delivery. As you know, research on bioactives has been the center of attraction nowadays, and 80% of the world population still depends on the traditional medicines that are based on the bioactives. And these bioactives are derived from different origins such as plants, animals, fungi, and bacteria. So these bioactives that are derived from different regions are of different types. These are lipids, terpenoids, alkaloids, phenolics, and carbohydrates. And one of the most common and most important bioactive is the carbohydrates, and these carbohydrates are composed of polysaccharide chains. So these are also known as biopolymers, and most common example of the biopolymers that are derived from the plants as plant exudates are mucilages and gums. And these mucilages and gums not only having the nutritional benefits but also having the medicinal benefits. So these gums and mucilages are used as an excipient for the controlled release of the tablets during the drug delivery system, and these are also used for making film coatings as well as oral films formulation, and these are also used for making shells of the capsules and for making solid dispersion. So these gums and mucilages not only having the nutritional benefits but also having the medicinal benefits, so can be taken as a green matrix for the metallic nanoparticle formulations to that are having the medicinal attributes. So these gums and mucilages are used not only in the pharmaceutical industries but also in the different food industries as an additives. So one of the common example of the mucilage is the date palm mucilage, and date palm mucilage that is derived from the dates of the date palm, and this date palm mucilage having somewhat sticky or gummy appearance with the dark brown coloration and having the smell of the glucose. And can be taken as an excipient for the drug tablet formulations. So the current project was planned to biosynthesize the silver nanoparticles that was capped with the green matrix of the date palm mucilage. And then further, these biosynthesized silver nanoparticles were further evaluated for their potential as a biobinder or as an excipient in the pills or tablet formulation for the better release of the drugs in the colon system. In order to achieve these goals, first step was to procure the mucilage sample from the local market. So the sample was collected from the local market and then further identified and then subjected to pre-treatment by washing and drying to treat from moisture or dust particles, and then further subjected to purification by using ethanol as a solvent. And then this purified sample was further subjected to proximate analysis by analyzing their moisture contents, crude fats, crude proteins contents, as well as determination of the mineral contents by analyzing ash of the mucilage sample. And these were also analyzed, evaluated for their antioxidant activity and antioxidant potential. For this purpose, different antioxidant assays were performed, that was diphenylbutyryl hydrazine free radical scavenging activity as well as total phenolic contents determination. These purified mucilage sample were further subjected to modification by carboxymethylation as well as nanoformulations. So after the carboxymethylation as well as nanoformulation, these samples were further analytically characterized by using different analytical techniques such as zeta sizer, UV visible spectrophotometric analysis, infrared spectrophotometric analysis, X-ray diffraction analysis, as well as scanning electron microscopic that was coupled with the electron dispersive X-ray spectroscopy. And after the evaluation, analytical characterization, these modified samples were further evaluated for the potential, for their medicinal potential, and for this purpose antimicrobial activities was determined, and these modified samples were further analyzed to evaluate their toxic potential towards human blood cells. For this purpose cytotoxicity assays were performed, and then these purified as well as modified samples were further evaluated for their pharmaceutical potential, and these modified samples were taken as an excipient for the tablet formulation, and these tablet formulation was further used for the determination of the disintegration time of the drug present in the tablet. That was bound with excipient date permissibility as an excipient, and then dissolution rate of these drugs were also evaluated. The sample that was taken from the market, it was dark brown in coloration, having some adhesive properties, and this with the smell of the glucose, and this sample was further subjected to purification by using ethanol as a solvent. After the purification, sample was powdered and having some light brown coloration. This purified sample was further subjected to carboxymethylation, and after carboxymethylation, its surface became somewhat smooth and with the greenish tinge. Whereas this purified sample was also subjected to nanoparticle formulation, silver nanoparticle formulation, or was subjected to nanoparticle formulation, which this mucilage was taken as green matrix. And then after the silver nanoparticle formulation, its color was dark brown. Its color appearance was dark brown. And this sample nanoparticle formulated date of mucilage was further subjected to nanoparticle size determination, and then it is clear from this figure that the size of the nanoparticle was 139.7 nanometer with the 100% intensity. These samples were also subjected to UV visible analysis. It is clear from this figure that an intense peak was appeared at the wavelength of 406 nanometer as a result of the reduction of the silver ions to metallic silver. In case of the silver nanoparticles, nanoformulated mucilage. And this peak became more prominent as the time was progressed. Whereas these peaks are not appeared in the crude or purified date palm mucilage. FTA analysis was also performed by these samples, crude, purified, as well as nanoformulated date palm mucilage, and the comparison graph is given here. From this comparison graph, it is clear that this graph indicating the some appearance and some disappearance of the functional group in case of the crude, purified, carboxymethylated, as well as nanoformulated date palm mucilage. And here is the presentation of the comparison graph that was taken from the ICAZ spectroscopy, and it is clear from this figure that which gives the correlation among the purified date palm mucilage, silver nanoparticles, silver nanoformulated date palm mucilage, and date model drug, as well as formulated tablet. X-ray diffraction analysis was also performed for the crude, purified, as well as nanoformulated date palm mucilage, and it is clear from the this and Oxide analysis was performed for the three-dimensional structure of the synthesized silver nanoparticles that was kept with date palm mucilage, and Oxide spectra of the crude mucilage displayed peaks exhibiting non-crystalline behavior of the sharp diffraction peaks, which is the indicative of the amorphous nature. As the spectra of the mucilage after purification showed the same amorphous nature with the no diffraction peaks, and diffractogram characterized little good, but this spectrogram is little good as compared to this crude mucilage. And the emergence of the minute peak in case of the modified mucilage is the due to the some order arrangement of the different particles in the modified date palm mucilage, showing some crystallinity behavior in case of the modified date palm mucilage. Here is the presentation of the comparison spectrogram of the silver nanoformulated as well as purified date palm mucilage. It is clear that in case of the silver nanoformulated mucilage, two peaks, prominent peaks were appeared at the diffraction angle of the 38.07 as well as 44.28 degree, exhibiting the face-centered cubic structure, and this match software was used to elucidate the arrangement as well as orientation of the diffraction angles, and correlation of these diffraction angles with the original JCPDS standards were done, and the correlation was analyzed up to 84.44%. These samples were also analyzed by scanning electron microscopy for the determination of their surface morphology. It is clear from this figure that the image of the crude mucilage shows some plain and smooth surface, whereas purified mucilage shows some less smooth surface, indicating the some amorphous behavior. Whereas when the sample was subjected to carboxymethylation, and then highly porous surface was appeared. Pores were appeared in the surface, and then silver nanoformulated mucilage showed the some less porous structure bearing minute particles of the round and uniform shape, indicating the embedding of the silver nanoparticles on the green matrix of the date palm mucilage. And these mucilages were further taken as an excipient for the drug formulation, and then their release in the different system of the body at different pH, and the tablet of the levofloxacin hemihydrate were made by using crude and nanoformulated mucilage as an excipient, and drug release in gastric medium having acidic pH. In gastric medium having acidic pH was observed. It is clear from the figure, that both excipients retard the release of the drug in gastric medium, and drug retardation was detected up to 14% for the drug bound with the nanoformulated mucilage. Whereas crude mucilage found the drug retardation was up to 33% in case of the crude mucilage. In enteric medium, having the pH of the 6.8, drug bound with the nanoformulated mucilage displayed regular release of the drug at the end of the 11th hour. So it is concluded from this all research work that this date palm mucilage as well as its modified form can be taken as an excipient for the release of the drug, or in other words, can be used as an excipient to delay the onset release of the drug in the acidic as well as basic media. So thank you very much.
Thank you, Dr. Fozia Anjum, for your wonderful talk. Our next presentation is by Sana Saeed Ahmad from University of Balochistan. She is going to present with title Antibacterial Activity of Silver Nanoparticles Against the Locally Isolated Strain of E. coli, a food pathogen. Please, Sana Saeed Ahmad.
Bismillahirrahmanirrahim. My name is Sana Said and my presentation is about the antibacterial activity of silver nanoparticles against a locally isolated strain of E. coli, a food pathogen. It was my MPhil research work. First of all, the introduction. Nanotechnology is the formation, composition, and handling of nanoparticles or elements with the size ranges from 0.1 to 100 nanometers. It provides new applications in many fields of science like molecular biology, genomics, and forensic, etc. It is used in prevention, diagnosis, and treatment of different diseases, water treatment, and in drug delivery. In this study, silver nanoparticles will be used as antibacterial agents for analyzing their antimicrobial activity against E. coli. Significance of study. In this research, silver nanoparticles are being used to demonstrate their antibacterial activity against E. coli. E. coli causes several diseases such as enteritis, septicemia, foodborne diseases, and urinary tract infections. These infections or diseases are of concern in the public health field. Silver nanoparticles can authenticate to be a substitutional method for the treatment of these diseases. In future, silver nanoparticles can be used to treat different foodborne bacterial infections, many skin infections, and for many diseases. Methodology. The methodology which I have used in my research work can be divided in 4 steps. Step 1, culturing of bacteria or E. coli. Step 2, identification of bacteria. Step 3, synthesis of silver nanoparticles. Step 4, estimation of antibacterial activity of silver nanoparticles on E. coli. Step 1, culturing of bacteria. For culturing purpose, streaking technique with the help of loop was used to isolate a pure strain of E. coli in the lab. All apparatus which I have used was sterilized. Eosine methylene blue agar or EMB agar was used as medium for E. coli. Figure shows the streaking method and the E. coli colonies on EMB media after incubation of 24 hours. In picture 1, streaking method or streaking pattern is showed, and in picture 2 or picture B, we can easily see the green metallic sheen or green colonies of E. coli. Step 2, identification of bacteria. E. coli was identified by the presence of green metallic sheen which we can easily see. For further investigation, gram staining was performed and slide was observed under the microscope. Picture shows the results after gram staining. Step 3 was preparation of silver nanoparticles by Turkovish method 1951. I have used this method for preparation of silver nanoparticles. Silver nanoparticles of 3 different concentrations, 1 millimolar, 3 millimolar, and 5 millimolar were synthesized. For 1 millimolar, 100 ml of silver nitrate, 1 millimolar solution was heated with the help of hot plate, and 11 ml of trisodium citrate, 10 millimolar solution was added dropwise till the color of solution changes into pale yellow or golden yellow. For 3 mm, 100 ml of silver nitrate, 3 millimolar solution was heated. 10 ml of trisodium citrate, 10 millimolar solution was added dropwise till the color of solution changes. For 5 millimolar, 10 ml of silver nitrate, 5 millimolar solution was heated with the help of hot plate, and 10 ml of trisodium citrate, 10 mm solution or 10 millimolar solution was added dropwise till the color turns into yellow. Characterization of silver nanoparticles. Characterization was performed by using UV-vis spectrophotometer. By using UV-vis spectrophotometer, we can easily see the peaks which our particles shows, and we can also estimate the size of particles. UV-vis of 1 millimolar silver nanoparticles is shown below. Figure A shows UV-vis peak of for the calculation of size, and figure B shows spectrum of monodispersity of silver nanoparticles. We can easily see the absorbance on picture B. UV vis of 3 millimolar silver nanoparticles. This picture shows the UV vis peak of 3 mm silver nanoparticles, and picture B shows spectrum for monodispersity of silver nanoparticles of 3 millimolar solution. UV vis of 5 millimolar silver nanoparticles. This picture shows the peak and monodispersity pattern of silver nanoparticles of 5 millimolar. We can see the peak which our particles showed and the absorbance which our particles have. Step 4. The last step was the estimation of antibacterial activity of silver nanoparticles on E. coli. After having the pure colonies of E. coli and preparation of silver nanoparticles, E. coli was treated with silver nanoparticles on Miller-Hinton agar. For this purpose, I have used well diffusion method, and I have also used 2 antibiotic discs for control, and one well was of water for a control. Inhibition zones showed by antibiotics and 1 millimolar, 3 millimolar and 5 millimolar silver nanoparticles can be seen in picture A, and picture B shows the zones showed by 1, 3 and 5 millimolar silver nanoparticles. We can easily see the zone of inhibitions around the wells. So it was all about my research and presentation. Thank you for your attention.
Thank you, Sana Saeed Ahmed, for your nice presentation. Our next talk is by Dr. Ghulam Hussain from Government College University, Faisalabad. Dr. Ghulam Hussain is going to present his work with title Peripheral Nerve Injury and Muscle Functions Restoration, a comparison of the various extracts of cannabis sativa. Please, Dr. Ghulam Hussain.
Hello everybody, here is Dr. Ghulam Hussain from Department of Physiology, Government College University, Faisalabad. I got my PhD from University of Strasbourg, France. Before starting my presentation, I would like to pay thanks and attributes to the organizer of this event, International Conference on Applied Zoology 2021. That's really an amazing platform that provides an opportunity to the scientists of not only the Pakistan, but the international community to share their knowledge and to share their latest work with the students, with the researcher, and with the audience. Really, that's going to help the people to learn more and more. So today I'm going to discuss my topic, and that is the peripheral nerve injury and muscle function restoration, a comparison of various extracts of the cannabis sativa. Before going into the depth of my presentation, I would like to introduce the topic, as it is known to everybody that our nervous system is the master command and control system that controls the function of nearly all other organs of the body. Particularly when we talk about the muscles, the information are obtained by using a complex network of the wire of the peripheral nervous systems, and the normal functioning of the muscle rely on the continuous supply of impulse by coming from these nerves. In case of any injury, or problem that may be due to the shotgun leg lubrication or or road accident, there is an interruption of the message or impulse from the nervous system to the muscles. And in the absence of these impulse or the signal, the muscles become functionless or their function are compromised. After an injury, there is a complex cascade of the event that starts just after an injury, and there's a process of cellular regeneration, clearing out the material, then sprouting, and then the establishment of the new connection. Although this process looks very simple, but this process is not very as simple as it looks. And if we summarize all these events in one figure, we can say that the process after an injury, there's a process of cellular regeneration, that the distal segment of the nerve is degenerated, then the clearance of the material debris, and then the sprouting of the proximal end, and then the re-establishment of the new connection, and then the functional restoration. But in nature of the process does not happen in this way, because there are many other factors and many other mechanisms that cause an hindrance toward the normal regeneration and the functional restoration. Then where we stand to treat such kind of the issue, because in Pakistan the number of road accidents are increasing day by day, and the people after such injury, they become bedridden for the whole life, and they become a function of social and economical burden, not only for their family but on the whole country. So what we do to treat such kind of the people? So here are the available strategies. For example, there are the symptomatic treatment, there are surgical approaches, supportive therapies, and medicinal approaches like gene therapy, stem cell therapy, and etc., etc. And there are some limitations associated with this treatment, because they are time-consuming, expensive, unavailable of donor, risk of immunosuppression, and 100% recovery is always a dream. So we can summarize that the medicine promoting functional restoration after such kind of the injury, they are still lacking. What can we do to overcome this issue? The medicinal plant and the bioactive compound, the biomolecule can be a source to help us in this matter. So we can say that the biomolecules or the bioactive compound, they have the health promoting effect, for example, antioxidative, anti-inflammatory, analgesic, antimicrobial, etc. So they might be helpful in accelerating the peripheral regeneration after an injury. Following this idea, here we follow a strategy. We screen out our medicinal plant that in this hope that any of them can help us to find out a good candidate that would really accelerate the rate of regeneration and functional restoration after an injury. To achieve this goal, and we follow this complex protocol of a work plan of a laboratory, although this is really difficult to explain everything in this short time. But interested people can contact us if they want to understand and they want to know more about this work plan. So by following this work plan, today I'm going to report the effect of various extract of cannabis sativa. This plant is well known. It's known to everybody due to its health promoting and other effects on our nervous system and on the other body organs. So based on the available data, one can speculate that the cannabis sativa can accelerate the motor function recovery after an induced mechanical insult to the peripheral nerves. To have an insight on this aspect, we use an animal model of sciatic nerve injury. In this model, we try to induce the peripheral nerve injury by making a compression insert to the sciatic nerve, and after this, we follow different parameter of this parameter to assess the rate of functional restoration that are lost after an injury. These parameters include food intake, body analysis, body weight analysis, walking track analysis, grip strength, and the response of thermal and the nociceptive stimuli. And following these parameter analysis, we found the results that reports that by including the various extracts, including the anaxin and ethyl acetate, their inclusion in the food did not have an effect on the eating behavior of this animal, and then the body weight remained also unchanged, indicating the unaltered metabolic state of these animals. And at the next step, when we measure the grip strength after an injury, we could observe that the both treated animal, they have better improvement in the functional restoration as compared to the untreated animal. And at the same time, we could also observe that the latency period toward the thermal and the mechanical stimuli, it was also in decrease in the treated animal, indicating that these animals have the improve in response to these kind of the stimuli. And at the next phase, we could also see that the treated animal, they have increased antioxidant capacity, indicating that these plants have some antioxidative effect on this animal. And at the next stage, the treated animals show that reduced oxidative stress. Next, we observe that the muscle mass after treatment that was really improved, and the rest were indicating that there was a corrected muscle atrophy after an injury. And next, we could see that there was some anti-glycemic effect of this plant, indicating that there may be some effect of this plant on the insulin-like receptor. And then the histopathology indicate that there was the surface area of the muscle fiber was also increased in the treated animal. So if we summarize all these things in our conclusion, that we can say that the cannabis sativa appears to have the potential to accelerate the functional recovery of following peripheral nerve injury, and we can say that the oxidative stress combating effect of this plant can be assumed to be the positive factor behind this speedy functional recovery. In future, we can think, and we are interested to do that the complete characterization of this plant, and that will lead us to identify potent bioactive molecules that will the actual player behind this effect, and what is the actual mechanism that is over there to increase the functional restoration after the treatment with this plant. That will be also interesting to dissect this thing. Dissect this thing. Based on these observations, we can say that the cannabis sativa can be a potential candidate to be identified and to be investigated in detail in the future study. And thank you very much for your kind attention. If you have any question,
Thank you, Dr. Ghulam Hussain, for your wonderful talk. Our next presentation is by Mohsen Ali from Kohart University of Science and Technology. He is going to present his work with title Biocontrol of Disease Caused by Melidogen incognita in Okra Plant using plant growth promoting Bacillus species. Please, Mohsen Ali.
Assalamu alaikum. My name is Mr. Mohsen Ali from Department of Microbiology, Kohart University of Science and Technology, Kohart. My research was on biocontrol of disease caused by Melidogen incognita in okra plant using plant growth promoting Bacillus species. Content: introduction, rationale, objective, material and method, result, conclusion, and differences. Introduction. Okra, locally known as bhindi, is a member of family Myrtaceae and a rich source of vitamin, minerals, and protein and carbohydrates. The okra crop is grown in 15,500 hectare areas, total production of 1,17,900 tons, and its yield decreased due to the disease caused by root knot nematodes. There are 23 species of nematodes associated with okra in Pakistan. Among them, Melidogen incognita is the most destructive one, which causes huge economic losses to okra production. Nematicides are generally used to control nematodes. However, they have several adverse effects, including toxicity, environmental contamination, and hazards. Plant growth promoting rhizobacteria have shown considerable potential as a biocontrol agent. Bacillus species reduce population density of soybean cyst nematode in greenhouse, microplot, and field study. The rationale of the study was the present study was designed to determine the plant growth promoting traits and the nematocidal potential of Bacillus species isolated from the field of district Kohart for the management of root knot nematodes, Melidogen incognita, in okra plant. The objective of the study was to isolate root knot nematodes from the root of infected okra plants, to isolate, identify, and evaluate plant growth promoting traits of Bacillus strains, to screen the selected Bacillus strain against Melidogen incognita using in vitro assay. Material and method. Soil samples were collected from 3 different locations in district Kohart. Sample was processed in water bath for the isolation of spore-forming Bacillus species. Next, serial dilution of soil sample were prepared, and then isolation of PGPR on LB media. Bacillus species was identified based on their morphological and biochemical identification. In morphological identification, colony morphology and microscopy was performed, and different biochemical tests was performed to identify the Bacillus species. Then PGPR trait of Bacillus subtilis were identified. Isolation of Melidogen incognita. Isolation of Melodega incognita from the root of infected okra plants. Then Melodega incognita were isolated by Berman-Fennell and root incubation method. Melodega incognita were then identified by morphological and materiological characteristics. Then identified Melodega incognita were propagated on okra plant grown in greenhouse. In vitro activity of Bacillus subtilis against Melodega incognita. Preparation of Bacillus subtilis culture broth, cell pellet, and supernatant was performed. Then disinfection and preparation of Melodega incognita. And then in vitro activity was performed by 2 ml of culture broth, cell pellet, and supernatant were transferred to the Petri plate containing 50 juvenile and 50 eggs per ml. Then statistical analysis was performed. Total 3 soil samples were investigated for the isolation of Bacillus species. I have isolated Bacillus subtilis bacteria, and all the 3 samples were positive for the Bacillus subtilis. These pictures shows the different biochemical test results that I have performed for the identification of Bacillus subtilis. I have performed different type of biochemical tests for the identification of Bacillus subtilis. This table shows different type of biochemical tests. Drum staining, spore staining, catalase, starch hydrolysis, BP, cell diameter, citrate test, 6.5% initial growth at 37 and 65, nutrient gelatin, motility, indole, and urease. Then all the 3 isolates were tested for the PGPR traits, and all 3 Bacillus subtilis were positive for the plant growth promoting traits. This picture shows that the subtilis promote plant growth promotes by different mechanisms like zinc solubilization, phosphate solubilization, ammonia production, and indole acetic acid production. Then Melodega incognita were identified based on their morphological and materiological characteristics. This table shows different type of morphological characteristics that indicate that I have isolated Melodega incognita from the root of infected okra plants. Body shape, body neck, body length, neck length, scale length, etc. All these are morphological data. Then meteorological data of Melodega incognita was characterized. These meteorological data shows that I identified Melodega incognita from the root of infected okra plant. Then I have performed in vitro activity of Bacillus subtilis against Melodega incognita. Effect of culture broth, cell pellet, and supernatant of Bacillus subtilis on mortality of Melodega incognita. I have isolated 3 different strains of Bacillus subtilis, and all these 3 strains have shown effective activity against Melodega incognita. Culture broth show approximately 70% activity against juvenile mortality of Melodega incognita. And then cell pellet of the Bacillus subtilis show about 50% activity against Melodega mortality. Then supernatant was isolated from the Bacillus subtilis strains, and then supernatant was applied. The supernatant shows also effective activity about 60% against juvenile mortality of Melodega incognita. It indicates that all the 3 Bacillus subtilis have effective activity against Melodega incognita. Then isolates were used against egg hatching of Melodega incognita. The effect of culture broth, cell pellet, and supernatant of the Bacillus subtilis on egg hatching of Melodega incognita. The culture broth show about 50% activity against egg hatching of Melodega incognita. Cell pellets show about 30% activity, and supernatant show about 45% activity against Nanodesign incognita. Conclusion. It was concluded that Bacillus subtilis show nematocidal activity against Nanodesign incognita and produce nematocidal metabolites. It also promotes plant growth by various mechanisms like zinc and phosphate solubilization, ammonia and indole acetic acid production. References. Thank you for your attention.
Thank you, Mr. Mohsen Ali, for your informative talk. Next presenter is Dr. Syed Qasim Raza from University of Veterinary and Animal Sciences, Lahore. He is going to present his work with title Multiparametric Detection of Non-Cell Autonomous and Cell Autonomous Deaths in Cancer Cells. Please, Dr. Syed Qasim Raza.
First of all, I would like to thank the organizing committee for selecting my abstract for as oral presentation in this international conference on applied zoology. My topic is very new for most of the audience, and I hope it will be interesting and will helpful for exploring the new research domains for most of the people. And this topic is basically multi-parametric detection of non-cell autonomous and cell autonomous death in cancer cells. Myself, Dr. Syed Qasim Raza, and working as assistant professor in IBBT UAS. First of all, we have to know about the types of cell death. There are basically, they are programmed cell death and there is accidental cell death. So type 1 and type 2 are programmed cell death like apoptosis and autophagy. Necrosis is type 3 and is accidental cell death. We classify it as an accidental cell death. There are some other deaths which have been classified recently, and those are like atypical cell death, like mitotic catastrophe and entosis. Basically, this is the entosis to who we called non-cell autonomous cell death. And when we called it the cell death, is programmed cell death caspase dependent and caspase independent. Apoptosis is caspase dependent, and the other ones are like autophagic and necroptosis. When necrosis is programmed, then necroptosis is programmed cell death. So interestingly, this phenomena of cell-in-cell internalization or cellular cannibalism or entosis was first time studied by Overholzer and colleague in 2007, and they have said that when two cells are coming together so close to each other and they interact with each other, and after some hours one cell is going to be internalized inside the other cells like a phagocytosis. So in this cell there was one target cell, other was the host, and target was going to be invade inside the other cells, so host cells. But they confirmed their results by interestingly finding that these cells which were going to be internalized through this look like phagocytosis phenomena were not like phagocytosis, because these cells were the live cell, and when they co-culture green and red cell together of basically same cell lines like MCF7 or MCF10 breast cancer cell lines, so they found that these cell-in-cell structures in their cultured cells. And after going for imaging, they found very interesting facts that these cells were just not overlapping outside. The red cell was not overlapping on the green cell, but it was completely internalized inside this green cell. So this complete internalization was confirmed by the confocal microscopic x-axis and y-axis images, and then was also confirmed by electron microscopy that there is this circled area like seam cell is complete cell is inside the other cells, and this show that this is the nuclei of this host cells, and here is the nuclei of the nuclei of the guest cell, and here is the nuclei of the host cells. After knowing about this, they confirmed their results by using the ZVAD inhibitor, which is the inhibitor of apoptosis, BCL2. When you overexpress BCL2, it means you are going to overcome on the apoptosis, and combine together BCL2 and ZVAD. They found that there was no reduction in cell structures. So they confirmed their result that this process is independent of the apoptosis, and they can say that this is non-apoptotic cell death process. They also confirmed their results. The results were confirmed like cathepsin B substrate, lysotracker, LUMP1. LUMP1 is basically lysosomal associated membrane proteins. And they confirmed through beta-catenin, which was the marker of the cell boundary. So they found this big giant cell having this cellular structure inside by lysosomes accumulated like a ring-like structure. So they confirmed that this cell is completely internalized inside the cells, and that will die through the cell and cell internalization. And they also found that these red colored show that these cells are basically inside and they are died through the lysotracker and cathepsin B staining positive, and they are dying through the lysosomes. So they say that the death of this after cell and cell internalization, so death is the lysosomal cell death. They also found some interesting facts regarding the blocking and cadherine antibodies. They found that there are the E-cadherines or P-cadherines. The cadherines are basically involved during these cell-cell interactions. And when you are going to block these cadherines, so you are going to inhibit the cell and cell internalization. So they use some chelating agent like EDTA and EGTA with some E-cadherines. So they found interestingly that there is the reduction of cell and cell internalization. Also the same for the MCF10A cell lines. After using these E-cadherine and P-cadherine antibodies, they found that after 12 hours in control, they have the 30% cell and cell internalization, but after 12 hours when we're using the E-cadherine, so these results confirm that there is involvement of cadherine at the junction of the cell and cell internalization. The interesting thing other regarding this entosis phenomena was this, that the cell was sometime going to be released after internalization like this. This was cell inside, and after some time the cell was released, and after releasing the cell can divide and live happily. The same for the other cell, that when cell are internalized inside the other cell like this, these cell inside the cell can divide. So this was also a very interesting phenomena, that so when there are now, you can say there are three ways. One is the cell is going to be die inside the other cell, other is the cell is going to divide inside the other cell, and third one is cell is going to be released from the other cell. These results confirm that the cells which are internalized are healthy cells. So they also confirm these studies in human tumors. They stain the human tissues with different markers of their cellular boundaries and their nuclear staining. They found like this structure, if you see, this is the complete cell boundaries, and this is the complete cell boundaries of the internalized cell. This show that the cell which is inside is completely live cell, is not like debris or something else. For us, we did our results also using these facts, because at that time this mechanism was not understood what will happen to the host and internalized cells. So we also did this study, cell and cell internalization. We also found cell and cell structure and different cell lines. We found that most of the cancer cell line have these cell and cell structures. And we also confirmed our results by these XZ and YZ and YZ imaging. So different axes confirm that these cells are not just like floating cell, but they are just cells which are completely internalized inside the other cells. So just a little video that you can see from my study, that how the cells are going to be internalized inside the other cells. So this green cell will be internalized inside the red cell. The same population is the same, but we only colored them through different cellular markers. After knowing about this, so for us, there was issue that the cell were going to be sticky together. Cell were just sticky cells. By using the flow cytometry, we wanted to isolate the double positive cells, but there was the issue that cells which are sticky will also be double positive, and they can be isolated from the flow cytometry as a double positive cells. So this problem was solved by our laboratories there, and they have found one technique which was image stream flow cytometry. So in image stream flow cytometry was just new technique at that times, and we found very interesting result during this by using this technology. So for example, you have the cell death type 1 and type 2 apoptotic, mitotic, pyroptosis, and autophagic. By using an vaccine 5 and REC7 staining like markers, and the same for the flow cytometry. For us, flow cytometry, for example, was a little bit different from the flow cytometry, because in flow cytometry, we have the passage of the cell like double positive, single positive, but at the same time, there was the laser light which was putting on the cells like this, and we get the image of these cells which are going to pass. So you have the passage of the cell, and at the same time, you have the imager which is going to take the images of the cells. So you can confirm your results that either these double positive cell, for example, here, if you see here different channels, green channel, red channel, and then the blue channel, and like the orange channel, and far red channel, and all are going to be submerged. And if you see here, this is green cell which is going to be internalized inside the red cell, the same double positive cell that we called it non-cell autonomous death. And these cells show double positive, and this by flow cytometry, and also were confirmed by these cells were confirmed that the cell were completely internalized inside the other. We did also some experiment on irradiation, so by using the radiated cell, and we found that the radiated cell were going to engulf the basically cell which were not irradiated, and we found their population in our experimentation. So due to we also did some experimentation on some inhibitor, and we confirmed that our cell and cell study was the same as like previously, that the cell death was non-apoptotic, and target cell percentage was cell which was internalized was degraded through the lysosomal cell death. So this image stream flow cytometry have multiple applications like cell signaling. You can study internalization, surface intracellular colocalization, DNA damage and repair, microbiology, cell cycle, cell death and autophagy, shape, stem cell biology, parasitology, cell-cell interactions. So you can have the flowing of the cell, you can have different color of those cells through flow cytometry, and at the same time, you have the imaging of these cells. So thank you very much for your attention, and thanks for, again, I would like to thank all the people who are listening to my lecture, and thank you very much.
Thank you, Dr. Syed Qasim Raza, for such an informative talk. Our next presentation is by Faryal Khan from Kohar University of Science and Technology. And the title of her presentation is Minced Meat Preservation by Using Gelatin-Based Iron Oxide Nanoparticles. Please, Faryal Khan.
Bismillahirrahmanirrahim. My name is Faryal Khan, and I am from Department of Microbiology, Kohar University of Science and Technology, Kohar, Pakistan. The topic of my research work is minced meat preservation by using gelatin-based iron oxide nanoplexus. So these are the content slides which I will discuss one by one. So let's have a start with introduction slide. So meat and meat products are perishable food products having a very limited shelf life, but it is a rich source of protein. So it is a good source for microbial growth, which result in food poisoning. So we preserve our meat by using different chemical preservative, which can increase the shelf life of meat. But due to consumer awareness, the use of synthetic chemical additive preservation by natural resources is getting popularity. So here I preserved my meat by using iron oxide, gelatin-based iron oxide nanoparticle, because iron is important function in biological processes, and it shows great potential as antimicrobial agent due to its controlled size, low toxicity, and high magnetism. So iron nanoparticle, what they do, they released the ions in iron oxide nanoparticle, they released and attach to the bacterial cell and result in protein denaturation and eventually cell death. The rationale of my project is that we propose to prepare iron oxide nanoparticles by gelatin. So gelatin here acts as a reducing and a capping agent, and we propose that iron oxide nanoparticle exhibited antimicrobial activity against foodborne pathogen. So we expect to circumvent the use of the toxic preservation for preservation of meat. The objective of my research is to evaluate the inhibitory effect of gelatin iron oxide nanoplexus against the strain that are Staphylococcus, E. coli, and Pseudomonas aeruginosa. And the last objective of my research is to investigate the preservative effect of gelatin iron oxide nanoplexus on beef. The material and method here, I used the 15 food samples that were collected from bakery store in district Kohar, and then I did cereal dilution and then grown on mutant agar, and then incubated for 24 hours. And after that, I do the gram staining for further identification of my isolated of my strains, and for that I did microscopy and biochemical test. The biochemical test I will discuss in next slides. So the second step is to prepare the gelatin iron oxide nanoplexus. For this, I did mix the two salts that were iron sulfate and iron oxide, and then I add the gelatin and ammonia. So these are the process, and after that I did the characterization, which I mainly do the with UV and XDR and SAM technique. And the third objective now is to put iron oxide, is to add iron oxide on my isolated strain to identify the inhibitory minimum inhibitory concentration. The last objective is to preserve radio effect of gelatin iron oxide on beef. So here I took the meat source from the slaughterhouse, and then I coated with the meat with a different concentration like 1.5 and 0.5 like this, and then stored at 4 degrees centigrade for 16 days. And after 16 days, I took my sample for microbial examination. And now the result, I have taken the 15 food samples for the isolation of Staphylococcus, E. coli, and Pseudomonas by using the different biochemical tests. And in this, I concluded that the E. coli was the most predominant in bakery sample, followed by Staphylococcus and Pseudomonas. And these are the microscopic images of my isolated strains. And these are the biochemical tests that I will perform for the identification of my desired for my isolated strains. And the test that I will use was catalase, oxidase, citrate, coagulase, indole, and methyl red. And how I know that my nanoparticles were prepared, when I added the two salts together, the color become orange, and when I add the ammonia and gelatin, the color become black, which indicates that the preparation of my iron oxide nanoparticle. These are the characterization images of my nanoparticles. The first A graph indicate that the wavelength of my nanoparticle, and the reference is strain is 240 somehow. And second is the XDR pattern. So we know that the XDR pattern actually shows the crystal behavior, while the third image is the same micrography technique, which shows that the size of my nanoparticle, which is about 15 nanometer. These are the MSC of my desired nanoparticles against bacteria. So the zone of inhibition I will discuss is in our next slide, that is the graph. And here the E. coli was the most predominant, so they show the highest zone of inhibition at 9 mm zone. The Staphylococcus were 7 mm zone, and the Pseudomonas aeruginosa, the zone of inhibition was 7 mm. And the different concentration that I will use was 100 microliter, 50 microliter, and 25 microliter. So the 100 microliter show the best zone of inhibition. And these are the last slide where I prepared my nanoparticle for the preservation of the gelatin iron oxide nanoparticles. So what I concluded from this graph is that at initial date there was no significant difference between the sample of control and other treated sample. The total viral count in control significantly increased with the increment of a time period at 4 days and crossed in limit up to 7 CFU per gram at 16 days, while the treated sample retarded the growth of total viral count during the whole period of time storage. The sample with 1.5% treatment shows significantly lowered total viral count at day 8 and 16. And the conclusion of I concluded my research work is that on the basis of that, it suggests that the preparation of iron oxide nanoplexus using gelatin, which is a less toxic, inexpensive, and safe. And I further concluded that it is used as an alternative antimicrobial agent, and it has significant preservative property to extend the shelf life of meat. These are my reference state, and I am reference slide, and I end up my presentation with these references. So thank you for your attention.
Thank you, Faryal Khan, for your nice presentation. Our next talk is by Dr. Malik Saadullah from Government College University, Faisalabad, with title A Novel Distechionate Treats Inflammations by Modulation COX-2 and Inflammatory Cytokinase in Rat Liver Tissue from Renia distechia. Please, Dr. Saadullah.
Bismillahirrahmanirrahim. A novel distechionate treats inflammations by modulation COX-2 and inflammatory cytokinase in rat liver tissue from Renia distechia. First of all, introduction. Importance of plants in Islam. In Holy Quran, it is said, your God is that who has made different kinds of orchards and gardens for you. Those are colorful crops of Phoenix dictylifera, Olea europaea, and Ponica granatum. Olea europaea. Olea europaea, that is olive, has found many references in Holy Quran and Hadith. In Hadith, it is said, eat the olive oil and apply it on the body. It cured 70 diseases. Kalwanji. According to Holy Prophet, peace be upon him, a regular use of kalwanji can cure every disease except death. Here are some important drugs of plant origin. First of all, aspirin. Chemically, it is salicylate. It has analgesic, anti-inflammatory, and antipyretic activity and obtained from Salix alba. The second one is atropine. Chemically, it is alkaloid. It has pupil dilator activity and obtained from Atropa belladonna. The third one is cocaine. Chemically, it is terpene alkaloid. Medicinal property, it has local anesthetic activity and obtained from Erythroxylum coca. Importance of natural products in modern medicine. Compounds from natural sources such as plants and animals play a significant role in modern medicine. They provide a number of extremely useful drugs that are difficult, if not possible, to produce commercially by synthetic means. These include such diverse groups of compounds such as alkaloid, morphine, codeine, vincristine, vinblastine, taxol, atropine, and hyoscine. Plant collection, identification, and extraction. Renia distechia was collected from peripheries of Lahore, Pakistan in August 2019. The plant was authenticated as Renia distechia by Professor Dr. Altaf Ahmed Dasti, taxonomist, Institute for Pure and Applied Biology, Bahauddin Zakariya, University, Multan, Pakistan. The voucher specimen has been deposited in herbarium. Dried and grinded plant material of Renia distechia was extracted by using different organic solvents such as dichloromethane and methanol. Through a successive maceration and filtration, the filtrates were concentrated by using the rotary evaporator. Result of extraction. The part used was aerial part. 900 gram of product plant were used. Organic solvent used was dichloromethane and methanol. 12 gram extract obtained from dichloromethane and 19 gram of extract obtained from methanol. Different phytochemical test was also performed. The result of phytochemical screening shows that saponin, steroids, and triterpenoids, flavonoids, cardiac glycoside, and tannins were present, while alkaloids and anthraquinones were absent. Here is the Hplc spectrum. The Hplc spectrum shows that quercetin, gallic acid, paracomeric acid, and synapic acid are present in Brenia dystachia. Metal analysis of Brenia dystachia was also performed. It shows that many metals such as iron, manganese, potassium, zinc, sodium, and magnesium are also present in the extract of Brenia dystachia. Here is anti-inflammatory assay guided isolation. By using open column chromatography, we purify the compound by repeated column using different mobile phases. The schematic diagram of purification is shown here. We elucidate the structure of pure compound by using different spectroscopic methods. These are the spectra of our pure compound. Physical parameters. It was a yellowish powder. Quantity obtained was 9-10 milligram. The melting point was 175 to 185 degrees Celsius. UV lambda max was 230-360 nanometer. IR was conducted by KBR method. Proton NMR by using 600 megahertz. C13 NMR, EIMS, and molecular weight and molecular formula was obtained by HREIMS. Here is the chemical structure of distactionate. Biological activities. Different biological activities were also performed. First of all, wound healing potential of Brenia dystachia methanol extract by Axion wound healing model. 10% cream of Brenia dystachia methanol extract, 20% cream of Brenia dystachia methanol extract, and novel distachionate was used against the standard contractobex. The novel distachionate shows a significant result against the standard contractobex. The second one was burn wound healing model. Here is the same 10% cream, 20% cream, novel distachionate, and the standard contractobex was also used. The novel distachionate shows a significant result against the standard contractobex. Carrageen-Pa edema model. In this model, saline, Brenia dystachia methanol extract distachionate was used against the standard indomethacin. The novel distachionate shows a significant result against the standard indomethacin. Cotton pellet edema model. Same as the saline, Brenia dystachia methanol extract distachionate and the standard indomethacin was used, while the distachionate shows a significant result against the standard indomethacin. Histopathological result. Histopathological analysis of liver, heart, lung, kidney, and spleen is shown here. Liver shows a mild inflammation which was significantly reduced after treatment with Brenia dystachia methanol extract. No renal and cardiotoxic activity was observed. Here is a molecular docking. Molecular docking was performed for confirmation of target size. It shows confirmed that novel distachionate treats inflammation by modulation COX-2 and inhibited cytokinase in rat liver tissue from Brenia dystachia. Here are the results obtained from docking. DPPH radical scavenging and acetylcholine esterase inhibition activities was also performed. Brenia dystachia methanol extract and novel distachionate was taken against the standard. In DPPH activity, quercetin was used as standard and distachionate shows a significant result against the standard. In acetylcholine esterase inhibition activity, Acerine was used as a standard and distachionate also shows a significant activity against the standard. Conclusion. In conclusion, current study isolation of new compound as 2-amino-678 trimethoxy 4A 56789 hexahydro 2-hydroxybenzo aniline 3-al 3,4,5 trimethoxybenzoate from methanol extract of Brenia dystachia isolated compound showed a prominent anti-inflammatory and antioxidant activities, which gives a clue about its immunomodulatory attributes. Molecular docking studies show that this distachionate X inflammation by modulation COX-2 and inflammatory cytokinase in rat liver tissue. Further studies to standardize the methanolic extract and long-term toxicological studies are warranted to establish the therapeutic potential of this novel compound. Here is a schematic diagram of the activity of distachionate. Distachionate cure the acute inflammation and chronic inflammation. It does not affect the normal cells. Thank you for your attention.
Thank you, Dr. Malik Sadullah, for such an informative presentation. Our next presentation is by Mutahira Subhan from Cohort University of Science and Technology. She is going to present with title Control of Disease Caused by Phytophthora capsicum in Pepper Plant using the soil-borne Bacillus species isolated from Cohort, Khyber Pakhtunkhwa. Please, Mutahira Subhan.
Fourth International Conference on Applied Zoology 2021, organized by the Applied Zoological Society of Pakistan and Quaid-e-Azam University, Islamabad, Pakistan. Assalamu alaikum. I am Mutahira Subhan. I am field scholar, Department of Microbiology, Cohort University of Science and Technology. My research topic is control of disease caused by the Phytophthora capsicum in pepper plants using the soil-borne Bacillus species isolated from Cohort, Khyber Pakhtunkhwa. Contents of my presentation is introduction, rationale, objectives, materials and methods, results, conclusion, references. Introduction of research is Phytophthora blight disease is caused by Phytophthora capsicum, a widespread soil-borne fungal pathogen. It is responsible for the severe damage to pepper plants, mainly when the soil contains the excessive moisture after heavy rainfall or irrigation. Pepper capsicum annum or capsicum frutescens is in the economically significant and valuable cash crop belonging to the family Solanaceae. Pepper crops cultivated over an area of 1,776,000 hectares throughout the world, with the production rate of 7,182,000 tons per annum. Biological control can be a promising and sustainable alternative for in the effective control of Phytophthora blights disease due to their environmentally friendly nature, avoiding the phototoxicity and fungicide resistance. The rationale of my study is the present study is designed to evaluate the efficiency of the soil-borne bacterial isolate against the Phytophthora capsicum responsible for the Phytophthora blight disease in pepper crop. This study, imported as the application of the biological control agent, is less costly and cheaper than any other chemical methods. Moreover, biocontrol agents give the protections to the crop throughout the crop period and also suffer to the plants, environments, and in the person who applied them. My research work have three objectives. First one is isolation and identification of Phytophthora capsicum from infected pepper plant. And second one is isolation and screening of the soil-borne antagonistic bacteria against P. capsicum. And third one is assessment of in vitro, and in vivo assay to determine the biocontrol potential of the selected bacterial isolates against P. capsicum. Materials and method. First of all, I have collected two types of sample, and for a sample of fungus and second one is the sample of a bacterial species, rhizospheric sample. First sample collection, sample collection for P. capsicii. Sample collection infected plants, parts, for example, leaves, stems, and roots. Sample collected from in the different area of district Kohat, Pakistan. Sample proceeded to the Department of Microbiology, Kohat University of Science and Technology, and isolation of P. capacity through PDA media and SDA media. Identification of P. capacity through special carrot agar media and morphological characteristics. And the isolation and identification of bacteria, antagonistic soil-borne bacteria, is a serial dilution and LP media. Identification of bacteria through different biochemical tests and morphological based. In vitro assay, antagonistic assay against P. capsicii, and in vivo assay, the in vivo assay against the P. capsicii responsible for the disease in pepper plant for vegetable field and of Kohat. After result and methodology, the methods and methodology, the results of my research work is identification of P. capsicii. The first picture show in the identification through the microscopy, and the second picture show the identification of P. capsicii through the special carrot agar media. This table shows the morphological identification of P. capsicii through the microscopy. I have observed the asexual spore sporangia. The range of sporangia length is 44.7 micrometer to 53.1 micrometer, and width is 24 micrometer to 38.4 micrometer. And next, I have observed the shape of sporangia. Shape of sporangia is pear shape. Color of sporangia is colorless, have no color. Papillate pointed at the tip. Spores fall from the colony. Long pedicle stay attached in the base of the spores. This table shows the biochemical test for in the Bacillus subtilis bacteria species. I have isolated 7 different isolates S1, 2, 3, 4, 5, 6, and 7. I have performed different biochemical tests like gram staining, endospore staining, catalase, oxidase, starch hydrolysis, VP, citrate, NECL 6.5. The first 2 tests, gram staining and endospore stainings, show they all of positive and rod shape, and catalase test all positive, but S7 show negative, and oxidase, the first one is show positive, other 2 negative, and S4, 5 show positive, and last 2 show negative. And starch hydrolysis, the first one show positive, 3 show negative, and the last 3 show positive. VP test, first show negative, second show positive, and 3 to 7 show negative result. And citrate test, first one show S1 show negative, and all of show positive result. And NECL 6.5 for 2 different temperature incubation with 2 different temperature, 37 and 65 centigrade. The first 37 centigrade S1 show 37 centigrade and 65 centigrade temperature show negative result, and S2 show positive, both of positive, and S3 37 show negative and 65 show positive result, and S4 both of show negative result, and S5 37 show negative and 65 positive, and S6 show 37 show negative and 65 5 show positive result, and S7 and both of show negative result. The activity of Bacillus species, this is the chart, show the Bacillus activity of Bacillus species, Bacillus subtilis, the Bacillus subtilis show the 90.70% activity, and the Bacillus cereus, the other species of Bacillus, show 90.30%. In vitro and antagonistic assay against the P-capsicide Bacillus subtilis, the picture A showed the control group and the picture B show the activity group. And the picture Bacillus subtilis show 6 millimeter zones. And this one is the in vitro antagonistic assay of P-capsicide Bacillus cereus. The A picture show control group and the second one show activity, and Bacillus cereus show 11 millimeter zone. This is the flow chart of in vivo assay. First of all, broth formations, a nutrient broth for broth Bacillus subtilis, and the PDB broth for P-capsicide. The incubation period of B subtilis for 24 hours, and P-capsicide for 5 days incubation. After incubation, serial dilutions. Prepare 1 mole of diluted P-capsicide put into 50 ml distilled water, and 1 mole of diluted Bacillus species put into 50 ml distilled water. After serial dilutions, both diluted solution apply on the healthy plant, spray on the plants, and after 15 days, the Bacillus subtilis show the activity against the P-capsicide. And this is the in vivo assay. After the application of fungus Bacillus species, this is in this picture show the in vivo assay. The conclusion of my research, this study concluded that Bacillus subtilis show the activity against the P-capsicide, which also promote the plant growth. This is the references of my research. Thank you for your attention.
Thank you, Mutahira Subhan, for your nice presentation. Our next talk is by Dr. Malik Hassan from Government College University, Faisalabad. He's going to present his work with title Pharmacological Evidence to the Usefulness of Coumarin in Metabolic Syndrome. Please, Dr. Malik Hassan.
Bismillahirrahmanirrahim. It gives me pleasure to share my research finding on International Conference on Applied Zoology 2021. I'm grateful to the organizers for providing me this opportunity to discuss this online oral presentation. So the topic of my today's talk is pharmacological evidence to the usefulness of coumarin in metabolic syndrome. I declare no conflict of interest. If you look at the metabolic syndrome or non-communicable disorders, these are raising up and up. Like in developing countries, overall in the world, 35 million people are suffering from non-communicable disorders, including metabolic syndrome. And out of 35 million, 80% of the population belongs to developing countries like us. If you look at the data, the WHO shows that 1 billion adults worldwide are overweight and around 300 million are obese. IDF is also sharing that the 415 million people are globally had been living with diabetes in 2017 and would shoot up to 642 million by the year 2040. By WHO estimates, the prevalence of this disease, even in Pakistan, will be as high, has been recorded as high 9.8% in 2016 and is likely to increase in the future. So CMDs are the cardiometabolic disorders usually depict a cluster of clinical features like insulin resistance, abdominal obesity, dyslipidemia, and hypertension. Dietary habits and the lifestyle patterns play a major role in it to aggravate and to promote and to boost the development of cardiometabolic disorders in the society. So keeping all these in consideration, the coumarins, which are the group of secondary metabolites, belongs to benzopyrone family, and these are found in many edible plants like coca beans, cassia, cinnamon, green tea, peppermint, and celery, so on and so forth. Its concentration is also varying from 1 mg to 87 mg per kg, and based on the substitution on its lactone and benzene ring, the broad category of the coumarin has been divided into 4 groups. So current study has been designed to optimize the safety and effectiveness of coumarin for metabolic syndrome using high sucrose in combination with fat-induced metabolic syndrome, a model developed in rats. Study protocol has been designed and approved by Institutional Ethical Committee, and then these pretty animals have been used for different tissues and in vivo experiments. Metabolic syndrome was developed in rats. In vitro assays were carried out using the isolated tissues from these rats, and then using tissue organ bath assemblies and histological studies were carried out along with serum biomarker analyses to see the effect of this administration on metabolic syndrome. So the measurement of non-invasive blood pressure in rats were measured using cuff-tail plethysmography in conscious, anesthetized conscious rats, and this is the setup here. You can see the animal restrainer. Animals are usually restrained in this one, and the signals with the use of tail pulse pressure, the signals are transmitted into PALab, and the data is represented in a graphical manner where this this pulse stops, that the systolic blood pressure is thought to be start, and this reading represents systolic blood pressure. Initially, at the first step, oral glucose tolerance test was conducted. Here you can see a different time interval. And if you look at the different different groups in high-fat diet administered rats, the oral glucose tolerance shows much more glucose concentration compared to the metformin cost of control and the treatment groups of coumarin, 2 doses, 30 and 70 mg per kg. Similarly, their body weights were found to be possibly decreased in treatment groups at week 14th of the treatment with coumarin at the lowest concentration of 30 and 70 mg per kg. When the systolic blood pressure effect was categorized, here we can see at the week 14, it's a much more pronounced, which is similar to week 12, and the effect was found more pronounced in the treatment groups, and even in both treatment groups at the dose of 70 mg per kg higher dose and the lower dose, the difference was also found pronounced, showing a dose-dependent blood pressure lowering potential of coumarin in high sucrose and the high fat-induced blood pressure rats. Here, if we could see, the diastolic effect was found pronounced, but it was much lesser compared to its effect observed on systolic blood pressure. Similar trend has been also seen on mean blood pressure. Then the mean blood pressure was also found to be decreased with different doses of the treatment. If you look at when at the terminal day of the study, when the animals were dissected, the serum was isolated and the serum was used to assess the effect of the administration on ADP, tamarin levels, leptin, and HMG co-reductase. So we found pronounced effect on the treatment of both these treatments, 30 mg and 70 mg per kg of the coumarin, and in most of the cases, the effect has been found dose dependent, particularly in case of coumarin level, in case of adiponectin levels. However, a trend was found on the part of leptin and HMG CoA reductase. So again, highlighting its potential and possibly by influencing or having an attenuating effect on coumarin levels, on leptin reduction, and also in pronounced HMG CoA reductase activity is highlighting its role in metabolic syndrome. Then tissue reactivity experiments or the vascular reactivity experiments were carried out using isolated tissue organ bath assembly, where these tissues were isolated from these animals and were mounted in these tissue organ baths, and the pressure transducer were there to assess any change in the elasticity of the tissues. Similarly, the aortic rings were mounted, and the acetylcholine was used to see the intactness of the endothelium. Here you can see the clear difference that here the endothelium was found intact, but however, in this case, endothelium was found denuded. So these observations were further checked on graphic presentations. Again, this one is showing the complete intact endothelium, this is showing partial intact endothelium, and this is showing around dysfunction of the endothelium. Here in a graphical representation, when this was checked for vascular relaxant activity, it pronouncedly relaxed the low potassium-induced contraction, and this was found to be inhibited when reproduced in the presence of non-specific inhibitor TEA or specific 4-aminopyridine. Isolated tissue organ bath assemblies, vascular integrity experiments also showed pronounced beneficial endothelial reviving retrieval effects on the part of the coumarin. And when the pancreatic tissues were assessed, it showed the restoration of the islets of the pancreas, where we can see in the treatment groups pronouncedly compared to this disease one. Their vessels were also being looked into, and we found the better architect in treatment groups. When immunohistochemistry was carried out for beta cells, mass assessment, the beta cell mass were found to be restored, which was in line in its in vitro findings in only H&E staining. And when the hepatic tissues were assessed, the normal architect was found in the treatment groups compared to the disease groups. So to sum these up, these data suggest that the coumarin possesses antihypertensive vasodilator properties mediated through activation of voltage sensor potassium channels, along with its effects on the adipokines. Therefore, this study provides a sound rationale for the pharmacological basis of coumarin in cardiometabolic syndrome, and this study could have been used as a lead-on for future studies to develop it as a newer drug for the treatment of metabolic syndrome. This is just to acknowledge the whole group and some of our collaborators from the University of California, from King Saud University, and from Khan University, and also my own university, Government College University, Faisalabad. Thank you very much.
Thank you, Dr. Malik Hassan, for your wonderful talk. Our next presentation is by Muhammad Amjid Ali from Bahauddin Zikriya University, Multan. He's going to present his work with title Next Generation Sequencing Identifies Pathogenic Mutations in HGF, POU3F4, TACTA, and MYO7A in consanguineous Pakistani deaf families. Please, Muhammad Amjad Ali.
Assalamu alaikum. Have a good day to everyone. I welcome to all of you on 4th International Conference on Applied Zoology. It's Muhammad Amjad here, currently doing PhD in Zoology from Government College University, Faisalabad. And I have done this work at Institute of Pure and Applied Biology, Bahauddin Sky University, Multan. The topic of my presentation is the next generation sequencing identifies pathogenic variants in HGF, which is hepatic growth factor, POU3F4, which is neurotranscriptional factor, and TECTA, which is related with the tectorial membrane, and MY07A, which is related with the myosin proteins in consequence Pakistani-Deaf families. First of all, we will discuss about little bit the introduction. The congenital diseases. Congenital diseases, these diseases are basically attributable to the genetic causes. The incidence of the congenital diseases is known to be higher in families with contiguous marriages. The congenital diseases, there are a lot of numbers, but the final one and the more number, which are the congenital deafness. The congenital deafness, they are basically related with the contiguous marriages, and these contiguous marriages which we have investigated, they are related to the genetic causes, and these genetic causes reveal that there is prelingual severe profound deafness, and ultimately 1% to 2% of newborns throughout the world, and among them approximately 70% of the deafness is attributable to the genetic causes. The genetics of the hearing loss is extremely heterogeneous as to date. More than 120 genes are reported to cause non-syndromic hearing loss. It means that the hereditary hearing loss, which are similarly numerous genes which are known to be cause the syndromic hearing loss. In recent years, the next generation sequencing has been increasingly implemented in the genetic diagnosis of the heterogeneous diseases, and it reveals that there are pathogenic variants which including the hearing loss providing a high through output efficient approach, and to target the hundreds of the causative agents and genes which are related to the whole exome for the identification of pathogenic variants. In details, we will discuss about the material and methods. In material and methods, we have gone through the subjects and clinical evaluations. In subjects, in material and methods, the first one is the subjects and the clinical evaluations. Three different families which are consequence families which are collected from Muzaffargarh, Rajanpur and Okada, which are present in the province Punjab, Pakistan, with multiple individuals suffering from deafness. Written informed consent were obtained from all the participants and their parents before their enrollment in this study. Family members affected with the deafness were examined in the ear, nose, and throat, which are basically at the district headquarter hospitals by the medical specialist. And then clinical evaluations, including the complete medical history interviews, comprehensive physical examination, and pure tone auditometry. Then otoscopic examination was performed to exclude the hearing loss due to infections, trauma, and other environmental factors. The vestibular function was evaluated by medical history, inquiry, and behavioral testing. This study reveals that the approved by the Research Ethics Committee of Peking University at China. Then the next, the genetic analysis. In genetic analysis, genomic DNA was extracted from the peripheral blood samples of the enrolled subjects by using the blood minikit which was obtained from Creatine Shanghai, and targeted next generation sequencing was performed in the proband from each family, and they are including in the whole family the customized capture panel which was targeted the axonic region of 414 non-deafness associated genes in addition to the axonic region. The panel also included all intonic, intergenic, and non-coding regions containing variants that reported in 147 genes and 751 variants, and candidates pathogenic variants were defined as non-symptomic, including the variants and defined as the supply site and indels, variants with minor allele frequencies. Then the in silico tools nutrition tester that was also performed. Then the co-segregation of the deafness phenotype and the pathogenic variants was confirmed in participating the family members by PCR amplification. Then we have also revealed that by the Sanger sequencing, all these supplementary and the pathogenic variants which were related to the non-sense, mid-sense supply sites and the other variants as well. Here is a whole number of different family which was obtained from Okada, and the different patients they are including in this family. Here are the results. In results, we have discussed about the clinical characterization, identification of the pathogenic variants, and identification of second pathogenic variants. We will go in detail one by one. In clinical characterization, there are at least 7 subjects in family which were obtained from Okada, and they were performed as basic functions bilateral, prelingual, and severe to profound the sensorial hearing loss. All affected subjects, which were included in the Okada family, they affected the subjects in the family were born to parents with consequence marriages. No ear malformation, vestibular disinfection, and developmental abnormalities or symptomic symptom were identified in all enrolled subjects. Next one is the identification of pathogenic variants. The identification of pathogenic variants, they are targeted by the next generation sequencing of 414 non-deafness genes were performed on probands of 3 families. Homozygous variants which were related with the leucine, tecta, and myosin proteins and hepatic growth factor, they were identified as the candidate pathogenic variants in the proband of family 4. In family number 1, which was originated from the Okada, all candidates variants having minor allele frequencies lower than 0.0001 in the public database of genomic AD, and the categorized as likely to pathogenic based as guidelines. The third one is identification of second pathogenic variants in family. Okada Sanger sequencing performed and confirmed that homozygous variants which were related to the leucine and surface proteins, they are present in the tecta and the deafness in all participating members in family of Okada which was obtained. Here are some results which were obtained, and here we can see the control and the patient comparison. We can see here the thymine is replaced by the cytosine, and cytosine is replaced by the thymine. All these are factors which were elated in the normal person. They are changed by different variants. In another example, here is another change which was arginine, and it must be replaced by the guanine. Here is the whole different patients which were obtained from different families, and these variants they including and they are compared with the normal person and control, in which we can see here is the change in the variation. There are the variants which are obtaining from these families, and it causes the basic problem in the deafness. Next, we are going to conclude this presentation. Here is, in conclusion, we can conclude that hearing loss is one of the major disabilities worldwide, which is often induced by the loss of sensory hair and the spiral ganglion neurons in the inner ear of cochlea. Spiral ganglion neurons, which are related with the hearing loss, could be caused by the genetic factors, aging, chronic cochlear infection, infectious diseases, ototoxic drugs, and noise exposure. And genetic factors account for around 70% of the hearing loss. In the present study, we are reporting the genetic cause of the bilingual, severe to profound deafness in the 3 contiguous families through the targeted next generation sequencing approach. And we are also reporting that the consequence marriages that should be comparable, and if there is any endocochlear potential or in the cochlea reduced function, that must be replaced, and that must be in domain of our specific individuals. Thank you very much for your attention.
Thank you, Muhammad Amjad Ali, for your nice presentation. Our next talk is by Shamsullah from Kohart University of Science and Technology with title Contamination Profile of Aflatoxin M1 Residue in Milk Supply Chain of District Bannu, Pakistan. Please, Shamsullah.
Assalamu alaikum. My name is Shamsullah, and I am going to present my research work, which are on the basis of contamination of aflatoxin M1 residue in open milk supply chain of district Bannu. Aflatoxin are produced by fungi in a tropical and subtropical region of the world. Due to this factor, this toxin is mostly present in Mucus, Aspergillus, Nanus, and Aspergillus parasiticus. These fungi mostly form symbiotic association with the leguminous plant like maize, rice, barley, etc. First of all, my presentation of outline is the introduction, objectives, materials and methods, results, conclusions, recommendation, and preferences. First of all, my research outline is the introduction. Mycotoxin are secondary metabolites produced by fungi in tropical and subtropical region of the world. Now, mycotoxin are a wide family of aflatoxin, which includes subtype of B1, B2, G1, and G2, and which are classified as a serious and chronic toxin for animal and human respectively. When these toxins are consumed by the mammals like जितने भी grazing animals है, cow, buffaloes, etc. Then these toxins are converted by the hepatic microsomal mixed function oxidase system into AFM1, which are secreted through urine as well as milk. Now, International Agency for Research on Cancer recognize this aflatoxin B1 or the carcinogen A, means these toxins are classified in such carcinogen which cause cancer mainly in human beings. So, now, this aflatoxin causes severe damages into DNA, primary liver cancers, hepatic cirrhosis, hepatitis, gene mutation, chromosomal aberration, immunosuppression, and cellular transformation in human. Aflatoxin amount in milk varies from day-to-day milking individual. Now, it depends upon the grazing of an animals, then in which climate it becomes graze and it consumes different types of foods. Now, Pakistan uses 33 billion liters of milk annually, which makes the Pakistan fourth biggest countries in the world. Now, majority of milk is produced in the informal areas and are supplied means as a raw milk, which are supplied both urban as well as into rural shops. Now, for the identification of aflatoxin, different methodologies are used in which capillary electrophoresis, ultra performance liquid chromatography, tandem mass spectrometry, and high performance liquid chromatography. But out of these different types of methodologies which are used for the identification of aflatoxin, now ELISA is the most reliable, which gives more accurate as well as faster result. In Khyber Pakhtunkhwa, ये जो gap study है, in Khyber Pakhtunkhwa, there is no routine testing of open milk for aflatoxin level that can lead to alarming situation. Therefore, the present study is designed with the aim to determine the presence and to quantify the level of aflatoxin M1 residue in the raw milk samples of the study area. Now, this study will be helpful in providing basic information to Khyber Pakhtunkhwa Health and for Regulatory Authority to take strong action to monitor as well as to regulate aflatoxin M1 in open milk supply chain. Now, इस रिसर्च की जो ऑब्जेक्टिव है कुछ यो है to determine and evaluate aflatoxin M1 in market milk sample of the district. But now market milk samples, it's maybe dairy shops as well as milkman, etc. Material and methods में first of all है study area and milk collection. Second one है quantification of aflatoxin in milk in through ELISA and statistical analysis. इसके बाद है result aflatoxin M1 level in raw as well as processed milk samples. But मेरा जो काम है वो है raw milk पे, तो यहाँ पर हम processed milk की बात नहीं करेंगे. Milk sample will be collected from dairy farms, dairy shops, as well as milkmen of the milk. Here we processed 92 milk sample of dairy milk, 92 milk sample of dairy shops, and 92 milk sample of milkmen, in which positive samples varies 23, 22, as well as 21. Presence of aflatoxin की जो परसेंटेज है वो डेयरी फार्म्स में 26% है. Now it is present in dairy shops 23%, while aflatoxin M1 present in milkmen of Gawala of Dumela 22%. इसकी मिनिमम जो लेवल है वो यूरोपियन कमीशन जो है उन्होंने इसकी जो मिनिमम लेवल बताई गई है वो है 0.05. यहाँ पर जो आई है वो है 0.211. 0. मैक्सिमम वैल्यू लेवल है ग्राम पर लीटर वो है 0.832 और इसकी जो मीन है ग्राम पर लीटर वो बनती है 0.541. मिनिमम लेवल ऑफ डेयरी शॉप्स जो है वो 0.244 है और मैक्सिमम लेवल है वो 0.879 0.665 वो है मीन ग्राम पर लीटर। मिल्क मैन ऑफ डुमेल जो है उसमें जो मिनिमम वैल्यू है वो 0.198 और मैक्सिमम वैल्यू लेवल जो है वो है 0.6484 और 0.422। यहाँ पर मिल्क मैन वाली में जो इसकी जो परसेंटेज कम आई है वो इसलिए आई है क्योंकि मिल्क मैन जो है वहाँ पर डिफरेंट टाइप्स ऑफ मिक्सचर जो होते हैं मिक्सिंग करते हैं, इसी वजह से इसकी परसेंटेज जो है वो कम आती है। Aflatoxin M1 level in branded and non-branded milk sample. मेरा जो काम है वो है non-branded milk sample means which are not pasteurized, which are not processed. यहाँ पर डेयरी फार्म्स की जो BDL वैल्यू है मींस बिलो डिटेक्शन लेवल, वो इन सब की जो है वो नहीं आई है क्योंकि वो बहुत कम है और हमारे जो मिल्क सैंपल है वो इसमें शो नहीं होती। हमारा जो रिसर्च है वो इसमें जितनी भी डिटॉक्सिफिकेशन है उसमें 0.11 से लेकर 0.2 या 0.2 जो है जो मिनिमम लेवल है ना, पहले ग्राफ में मैंने शो किया था, इसमें डेयरी मिल्क में जो परसेंटेज आई है वो 8.7% है और डेयरी शॉप्स की जो आई है वो है 9%। इसी तरह मिल्क मैन जो है डोमेल वाली उसमें आई है 14%। और सेकंड वन जो कंसंट्रेशन है वो 0.21 से लेकर 0.3, इसकी जो परसेंटेज है टोटली 5 सैंपल है, 5 सैंपल में जो परसेंटेज आई है वो है 21% और डेयरी शॉप्स की जो 3 मिल्क सैंपल है उसमें 13% आई है और मिल्क मैन की जो 2 सैंपल्स है उसमें 9% आई है। लास्ट वाली में अगर हम देखें तो वो है 0.712 to 0.8 जो मैक्सिमम लेवल है। ऐसे जो पहले की टेबल मैंने बताई थी तो उसमें मैक्सिमम लेवल है पर ग्राम पर लीटर 0.8, तो इसमें हाईएस्ट परसेंटेज जो है इसी 2 मिल्क सैंपल में वो आई है 8.7% और डेयरी शॉप्स की जो है वो 1%, क्योंकि मिल्क मैन में ये परसेंट मिल्क मैन वाली जो सैंपल होती है और उसमें डिफरेंट टाइप्स ऑफ मिक्सचर पाए जाते हैं, तो इस वजह से इसकी जो हाईएस्ट नंबर है ग्राम पर लीटर वो शो नहीं की गई है। Now, milk is an essential source of, we know that, nutrition, and thus its consumption in increase parallel to continuous increase of human population. In Pakistan, particularly District Banno, data regarding aflatoxin contamination is scarce since no regular surveillance of mycotoxin exists. In this regard, this study is a reflection of overall occurrence level of aflatoxin M1 in raw milk, including dairy farms, dairy shops, milkmen, in the context of Pakistan, particularly District Banno. This study provides a complete profile of aflatoxin M1 present in non-branded milk sample collected from District Banno. This study identifies that non-branded milk samples as highly exposed to such a toxin compared because in processed milk, the milk is processed in 170 degrees centigrade for 3 to 5 minutes, in which this toxin becomes denatured. The presence of such a toxin in milk is a serious problem considering the extraordinary usage of milk and milk-associated product in Pakistan. Henceforth, this study provides a strong base for food and health regulatory authorities of Khyber Pakhtunkhwa, Pakistan to take firm steps for constant monitoring and regulating of such toxin in milk. Strict permissible limits should be implemented to avoid fungal contamination. To the best of this is the first study that examined the aflatoxin M1 detection through ELISA in non-branded milk sample, a concentration of aflatoxin in the context of particularly Khyber Pakhtunkhwa, Pakistan. Thank you. Stop recording.
Thank you, Mr. Shamsullah, for your informative talk. Our next presentation is by Ibrahim from Kohart University of Science and Technology. He's going to present his work with title Control of Nematodes, Nalidogen incognita in the common bean plant by Trichoderma species isolated from the fields of district Kohart. Please, Mr. Ibrahim.
Bismillahirrahmanirrahim. Assalamu alaikum. My name is Ibrahim and I am a student of the Microbiology at Kohart University of Science and Technology, Kohart. This is my presentation topic for abstract presentation is control of root knot nematode, Nalidogen incognita, in common bean plant by Trichoderma species isolated from the fields of district Kohart. Content of my presentations are introduction, rationale, objectives, material and methods, results, conclusion and recommendation, and references. Introduction. Common bean, Phaseolus vulgaris, is a member of the family Fabaceae. It contains low fats, high protein contents, dietary fibers, and complex carbohydrates. In Pakistan, due to various reasons, including nematodes infestation, the production of common bean is insufficient to meet the requirements. For the control of plant pathogenic nematode, Trichoderma species considered as a suitable biocontrol agent. Biocontrol reduces the reliance on synthetic pesticides, reduces the adverse effect on the environment, improves worker safety, and maintain the economic viability of crops. Rationale. Therefore, the present study was based on isolation of nematocidal strain of Trichoderma species from the fields of district Kohart against the root knot nematodes carrying disease in common bean plant. Objectives. Objectives are to isolate and identify the Trichoderma species from the fields of district Kohart, to extract and characterize root knot nematode from the root of common bean plant, and to investigate the nematocidal activity of Trichoderma species using the in vitro and in vivo bioassay. Material and methods. In material method, first of all, soil samples were collected from different locations in district Kohart. Soil would proceed. Then next, a cereal dilution were prepared. For the isolation of Trichoderma species, the cereal dilution were poured on two different media, potato dextrose agar and Trichoderma selective medium, and incubated at 25 degrees centigrade temperature for 3 days. After incubation period, the suspected Trichoderma colony were subcultured on PDA and were subjected for morphological identifications. The morphological identification were based on colony morphology and microscopic characteristics. Isolation of Nalidogen incognita. Root samples were collected from infected common bean plants. Then nematodes were isolated by the permanent method from the and root incubation method from the common bean roots. Then the isolated nematodes were identified morphologically and morphometrically. The identified nematode were then propagated on common bean plant grown in greenhouse conditions. In vitro and in vivo nematocidal activity of Trichoderma. During in vitro nematocidal activity of Trichoderma gonadia suspension and culture filtrate of Trichoderma were prepared. Then the nematodes suspension were prepared by disinfection it and prepare for the in vitro activities. Then 1 ml of gonadia suspension and culture filtrate transferred to a petri plates containing 50 juveniles or eggs per 10 microliters. During in vivo nematocidal activity of Trichoderma against root knot nematode Nalidogen incognita, Trichoderma gonadia suspension were prepared. Then the seed of common bean plant were sterilized and treated with Trichoderma gonadia suspension. The treated seeds of common bean were planted in a pots containing 1,000 nematodes per pots. After 8 weeks, The different plant characteristics were measured and then statistically were analyzed. Results. Total five soil samples were investigated for isolation of Trichoderma species. In all the samples were positive for Trichodermas. The figure 1 shows microscopic pictures and colony pictures of the isolates. The upper rows are the microscopic picture, while the middle and below rows are the front and back side pictures of Trichoderma isolate colonies. Out of 5 isolate, 3 were selected based on in vitro activity for in vivo and morphological and molecular identifications. Table 1 shows colony characteristics of Trichoderma isolates on PDA and SNA, synthetic nutrient agar. All 3 isolates have shown different characteristics on different media on PDA and SNAs. Characteristics such as color of the culture, colony, form of PDA, conidiation, and pigmentation were noticed. Microscopic characteristics of Trichoderma isolates. These are the microscopic characteristics of Trichoderma isolates. The microscopic characteristics such as shape and size of conidia, phyllides, presence or absence of striated head, and presence or absence of dermatospores were noticed. All the isolates were noticed to produce all the isolate were noted that the striated hair were absent in all the isolates, while dermatospores is present only in the isolate 2. Morphological and morphometrical data of Merodogan. This table shows the different characteristics of Merodogan and Cognata. And characteristics includes body shape, body length, neck length, stet length, knob shape, stet knob length. Table 3 shows different characteristics of Merodogan and Cognata. These characteristics includes body shape, body length, neck length, stet length, knob shape, stet knob length, interior into exterior fold, tail lengths of female and male Merodogan and Cognata. These characteristics were previously also reported by Carson et al. Figure 2 shows the Trichoderma conidia suspension and cultural rates on J2's motility. The highest activity of Trichoderma isolates were noted against J2 motility in the isolate 3. Conidia suspension shows 68% of the J2's motility, while in culture filtrates J2 motility were 71%, and so on. Figure 3 shows the effect of Trichoderma conidia suspension and culture filtrate on egg hatchings. The egg hatching inhibition, the highest egg hatching inhibition were noticed in isolate 3. Culture filtrate has shows 68% of the egg hatching inhibition, while the spore suspension has shows 58% of the eggs hatching inhibitions, and so on in isolate 1, isolate 3, isolate 4, isolate 5, and isolate 3 were the most effective. Figure 4 shows the effect of Trichoderma on galling and egg mass index in the common bean plants. These were the in vivo studies during which the highest activity of Trichoderma isolate 3 were noticed. Trichoderma isolate 3 has significantly reduced the gall index and egg mass index in common bean plant and has also shows the plant growth promoting traits. Figure 5 shows the antagonistic activity of Trichoderma against plant pathogenic fungi. The highest activity of Trichoderma isolates were noticed in isolate 3. The fungal pathogen were Aspergillus niger, Rhizoctonia solini, and Fusarium aureus. The highest activity against Rhizoctonia solini were noticed about 25%, while against Fusiorhizopodia, the activity of Trichoderma aspirate were 73%, and against Aspergillus, the activity were 58%. Conclusion and recommendation. From the study, it is concluded that Trichoderma isolate ISO3 has shows a significant toxicity against Mesogyna pugnata during in vitro and in vivo both studies. Further, a large-scale study is required to find the efficacy of above in the fields. These are the references. Thank you for the attentions.
Thank you, Mr. Ibrahim, for your nice presentation. Our next talk is by Dr. Syed Makhdoom Hussain from Government College University, Faisalabad. Dr. Syed Makhdoom Hussain is going to present his work with title Analysis of Growth Parameters, Neutralization and Body Composition of Catla fingerlings fed on Moringa oleifera leaf meal-based diet. Please, Dr. Syed Makhdoom Hussain.
Assalamu alaikum everyone. I am Dr. Syed Makhdoom Hussain, Associate Professor at Department of Zoology in Government College University, Faisalabad. And the topic of my today's presentation is analysis of growth parameters, nutrient utilization, and body composition of Catla fingerlings fed on Moringa oleifera leaf meal-based diet. First of all, as we know that aquaculture is one of the fastest food producing industry all over the world, and it is estimated that by the year 2030, the global aquaculture production will increase up to 109 million tons because of its rapid increase in the world population. Aquaculture accounts for 46% of the global production and 52% of all fish consumed by the human beings. All around the globe, high quality protein is available at reasonable cost that is supplied by the aquaculture industry. The aquaculture industry still faces a number of issues that threaten its long-term viability. All over the world, commonly fish meal is being used in the formulation of fish feed. Why we prefer fish meal? Fish meal is basically a major source of protein and essential nutrients in fish feed. It is highly digestible because it contains essential amino acids and minerals. But with the passage of time, increasing demand, unstable supply, and high prices of the fish meal make it necessary to search for the alternative protein sources. Basically, the main objective for most of the fish farmers is to produce high quality fish feed at low cost. With the increasing fish meal prices, the scientists are being motivated to identify the cost-effective alternatives of the fish meal. So when we see the alternatives of the fish meal, we find that plant byproducts are at number one. Why we prefer to use plant byproducts for the replacement of this costly fish meal? Because these plant byproducts are most promising sources of protein and energy for the aquaculture feeds. They are basically a biodegradable and cost-effective and helpful in developing the environment-friendly fish feed. Moringa oleifera was used in the present study. Basically, it is a famous tree known as a miracle tree because it has an impressive range of the medicinal uses. It is highly digestible and a very suitable amount of the protein content is present in it. We selected Moringa oleifera leaf meal to formulate fish feed. Moringa oleifera leaf meal improves the growth performance of the fish because it has important source of the vitamin and mineral. These leaves are rich in carotenoid, ascorbic acid, and minerals, especially iron. And it is a promising protein source at low, when we use these leaves at low levels. These leaves have high amount of the other essential minerals such as calcium, copper, zinc, potassium, phosphorus, magnesium, and sodium, etc. The crude protein content that is present in these leaves is about 25 to 32%. Catla was the fish that was used in the present study. It is basically an Indian major carp. It is commonly known as thella and being cultured all over the Pakistan, especially in the Punjab province. This fish is a surface feeder fish. It has high commercial value due to consumer choice. It is widely cultivated under polyculture system with other fish species such as the Labiohita and Sirenus mirigella. This fish has a high growth rate. Materials and methods. All of this research work was conducted in the Fish Nutrition Lab at Department of Zoology, Government College University, Faisalabad. Fish and experimental condition. The Catla fingerlings were purchased from the Government Fish Seed Hatchery, Satyana Road, Faisalabad. After procuring these fingerlings, these were acclimatized to experimental conditions in laboratory for 2 weeks in V-shaped tanks. During this period, the fingerlings were fed twice daily to apparent satiation on the basal diet that was used in the subsequent digestibility study. Aeration was provided around the clock to all the tanks through capillary system. Water quality parameters such as the water temperature, pH, dissolved oxygen were monitored on the daily basis during this whole study. Moringa oleifera leaf meal was formulated by processing its leaves. The Moringa oleifera leaf meal were collected from the southern Punjab area, especially from Multan. These leaves were then soaked overnight in a tank to remove the saponins and other water-soluble anti-nutritional factors like phytate. These leaves are placed on a wire mesh to drain excess water and then spread on plastic sheet to dry under the shade to avoid loss of the vitamins through photodynamic damage or oxidation. Then these leaves were threshed from the stalks to reduce the crude fiber content in the meal. These dried leaves were grinded into fine powder by using a hammer mill and stored in plastic bags at room temperature for the further processes. Feed ingredients and experimental diets. These feed ingredients were analyzed for chemical composition before the formulation of the fish feed. The reference diet was prepared to supply adequate levels of the required nutrients for the normal fish growth performance, while an inert marker like chromic oxide that was used as inert marker about 1%. Formation of the feed or pellets. These feed ingredients were finally grinded to pass through the 0.5 mm sieve size. All these feed ingredients were mixed by using a feed mixer for about 5 minutes, and fish oil was gradually added thereafter. Amount of water, about 10 to 15%, was added to prepare a suitable texture called dough. This dough was then processed by pelleting machine to make feed pellets. Here is a table in which you can see the composition of the test diets. In this table, you can see that 0%, 10%, 20, 30, 40, and 50% fish meal was replaced by the Moringa oleifera leaf meal. A review of feeding plate and fecal collection tube. While these are the valve 1 and valve 2, by opening valve 1 and valve 2, we collect the fish feces in this study. Sample study of feed, feces, and fish muscle. Crude protein was determined by the gel apparatus, crude fat by the Soxhlet apparatus, ash was determined by furnace, whereas growth performance such as weight gain percentage, specific growth rate, and feed conversion ratio was determined by these standard formulae, and digestibility was also calculated by this formula. Statistical analysis. Data of the growth performance, nutrient digestibility, and carcass composition of test diet was subjected to one-way ANOVA by using standard method. whereas the differences among treatments were compared and considered significant p less than 0.05. The co-stat computer package was used for the statistical analysis. Results in this table, we can see that growth performance of the Catella fingerlings at different levels of Moringa oleifera leaf meal based test types. In this table, we can see that fish fed on Moringa oleifera leaf meal based diet significantly improved the growth performance of Catella fingerlings. At 10% replacement level, the maximum weight gain was obtained. That was 20.64. The second best value of the weight gain, that is 18.56, were observed at 20% replacement level, while the minimum value of the weight gain 12.46 was observed at 50% replacement level. So this indicates that the maximum utilization of the leaves can be up to 10%, while when we try to increase the inclusion level of this meal, like Moringa oleifera leaf meal in plant-based diet, the growth performance started to decrease. At 10% replacement level, the lowest FCR 1.40 and highest specific growth rate 1.49 were observed, indicating the maximum growth and significantly different from all other groups. Maximum FCR, in other words, you can say the poor FCR 2.25 and lowest SGR 1.11 was observed at 50% replacement level. In other table, we found that comparison of mean of apparent nutrient digestibility coefficient of Catella fingerlings fed on Moringa oleifera leaf meal based diet. In this table, we found that a significant improvement in growth parameters was found, which marks an indication of the maximum digestibility of the nutrients such as crude protein, that was about 75.33, whereas crude fat 83.28, and the gross energy digestibility was 65%. In fish fed on Moringa oleifera leaf meal based diet at 10% replacement level. Maximum digestibility of the nutrients such as crude protein, crude fat, and gross energy were also observed at 10%, where a minimum amount was observed at 50% replacement level based diet. And about body composition of Catella fingerlings fed on varying levels of the Moringa oleifera leaf meal based diet, there was significant difference in terms of the crude protein, crude fat, ash, and moisture contents of the Catella fingerlings. The maximum value of crude protein, that is about 19.66%, was observed at 10% replacement level, and whereas the minimum value of the crude protein was observed at 50% replacement level. Crude fat 3.43% was observed, and moisture 74.43% were minimum at 10% replacement level, whereas these values were maximum at 50% replacement level, 5.38%. That was maximum amount observed in the present study. It was determined on the 50% replacement level based diet. Similarly, when we talk about the ash content, that was also highest, 2.47% at 10% replacement level based diet, whereas it was lowest, 1.44% at 50% replacement level diet. So on the basis of these results, it was concluded that Moringa oleifera leaf meal has a good perspective for to be used as fish meal replacement in Catella fingerling based diet, but only about up to 10% level, and whereas it is also cost effective without compromising growth parameters. It was also observed that it reduces the discharge of the nutrients into the water, resulting in environment friendly fish culture. But after this inclusion level, when we started to add this at 20%, 30%, 40 or 50%, the fish growth performance, fish nutrient digestibility, its body composition, all these parameters were going to decrease. So it is recommended that Moringa oleifera leaf meal should be used up to 10% level. I am thankful to Higher Education Commission Pakistan for funding this research work. Thank you. Here are some references. Thanks for your patience.
Thank you, Dr. Syed Makhdoom Hussain, for your wonderful talk. Our next presentation is by Habibullah from Kohat University of Science and Technology. He is going to present his work with title Synergistic Effects of L-N-Gutifolia Oil on Antimicrobial Activity. Please, Mr. Habibullah.
So my research topic is synergistic effect of Lavender angustifolia oil on the antimicrobial activity of gentamicin against methicillin-resistant Staphylococcus aureus. So my name is Habibullah, MPhil scholar, botany registration number, and my supervisor is Dr. Nisar Ahmad. Second supervisor is Dr. Saeed Ahmad. Department of Botany, Kohat University of Science and Technology. So introductions. Lavender angustifolia, botanical name of lavender plants. Lavender oil also used for research purpose and combination of many antibiotic like gentamicin drugs, other ones. So Lavender angustifolia is a common name of lavenders. Flowering plant and main family Lamiaceae. And these ones high altitudes, found in high altitudes and more than South Africa, Europe, and other ones, and Southeast India. So the uses of lavender oil are used to make many medicines. They also used for an anti-inflammatory, pharmaceutical product, cosmetic and perfumes, and justice, etc. So gentamicin drugs. Gentamicin is the antibiotic class of aminoglycosides. Gentamicin affects both gram-positive and negative bacteria. Gram-positive bacteria infection caused by Staphylococcus aureus. Gentamicins work stopping the growth of bacteria infection like endocardic pneumonia, urinary tract infection, skin infection, and sepsis, inflammatories, etc. So the synergistic activity, the combination of two compounds of good effect as compared to individual compounds. So alone one compound has not effective. So the combination of two compounds is more effective as compared to individual. So synergism checks between the gentamicin and missile antimicrobial agent like such as essential oils. So a synergistic activity was evaluated for the lavender essential oil and associated gentamicin drugs against MRSA and MSSA. MRSA methicillin resistant Staphylococcus aureus and methicillin susceptible Staphylococcus aureus. So MRSA and MSSA is a bacterial strains. Methicillin resistant Staphylococcus aureus is also called MRSA, and methicillin susceptible Staphylococcus aureus is also called MSSA. So there are some possible infections of Staphylococcus aureus are NQs. Skin infections, fever, gastrointestinal symptoms, fever, sepsis, pneumonia, endocardic, etc. So my research hypothesis, lavender essential oil and gentamicin drugs may possess a rich synergistic effect, which may lead towards a biological activity. My objectives: to study antimicrobial activity of lavender oils against MRSA and not in combination with gentamicins, And another objective and also investigate the kinetic growth of bacteria of gentamicin drug and lavender. Material and methods. So study area, my study area is also called, the study will be conducted in the Department of Pharmacy, University of Science and Technology, the MRSN assistant, KBRN 13, KBRN 23, and KBRN 31 will be previously collected by Pharmacy Lab cost. So antimicrobial activity agents. The antimicrobial activity of the lavender essential oil and gentamicin drug against the Paleococcus aureus and the synergistic effect of oil and with integrated with gentamicin were carried out by using the microdilution methods. So first of all, freeze the overnight culture will be placed on the surface of molar hinges plates uniformly. Plates will be incubated 37 centigrade temperature for 24 hours. So these, first of all, I determine the MIC by broth microdilution method. So these methods are used for determination of MIC of gentamicin drug and lavender oil according to the guidelines recommended by the Clinical and Laboratory Standards Institutes. So first of all, I using the methods of checkerboards. Checkerboard method was designed to combine effect of two agents for the purpose of combination. The both broth microdilution checkerboard also 96 film microtiter plates are used for the MIC purpose. So the results of checkerboards also determines and show for FIC friction and habitual concentration. So this one FIC index value represent the comparison of two compounds like FIC value index additive less than 0.5 and synergy is greater than point. A synergy is less than 0.5 and additive greater than 0.5. And different and 1 to 4 and antagonisms FIC value show the greater than 4. So synergism value also shows my results result 0.5 less than. So another methods assay are used for time catalysis. This time catalysis performed using same broth microdilution methods. So lavender oils in combinations with antibiotic gentamicin were tested against MRSA strains. First हम identify करते हैं colony forming units of MRSA strains. And after this, identify the combination of two compounds. जो compound होते like gentamicin and lavender oil are combined and determine the CFU colony forming unit for time 2 times. This one results, first of all, one strains concentration of drug MRSA KBR in 13. This key concentration, one wells में 96,000 है, and seconds well में 480,000 and third wells 24, so on. So first strains जो KBR in 13 इसकी जो MIC हमारे पास आई है वो है 12,000 माइक्रोलीटर पर ml. और इसके अलावा ये जिंटामाइसिन के MIC और ऑयल्स की जो MIC है 500 माइक्रोग्राम पर ml. ये वन स्ट्रेन के MIC है जो लेवेंडर ऑयल्स एंड अदर वंस दूसरा जो स्ट्रेन्स है उसकी MIC हमारे साथ ड्रग की जो है वो 24,000 MIC है और ऑयल्स की जो MIC है 23 KBRN स्ट्रेन्स पे जो है 250 इसकी MIC लेवेंडर ऑयल्स की है और जो थर्ड MSSA स्ट्रेन्स है इस पे जो MIC हमारे पास वो 1500 है ड्रग्स की माइक्रोग्राम पर ml और ऑयल्स की जो MIC है 62.5 ये लेवेंडर ऑयल्स की MIC है और हमारे साथ रिजल्ट जो है FIC बाय ब्रॉथ माइक्रोडाइल्यूशन मेथड. So first of all, MRSA strain जो है KBRN 13 इसकी एलॉन MIC 12,000 है और लेवेंडर ऑयल्स की एलॉन MIC 500 है और कॉम्बिनेशन जो है 1500 जो है 1500 इसकी MIC है और 125 जो है ऑयल्स की MIC है. جب اس کی کیلکولیشن ہوئی تو اس کی FIC 0.37 آیا تھی، تو اس کی ریزلٹ ہمیں سائنرجزم شو کرتے ہیں. اور MRSA کا KBRN 23 یہ جو دوسرا اسٹین ہے اس کی MIC الان 24,000 ہے اور آنس کی 250 ہے. تو اس کی جو وہ ہے FIC ہے 0.31 ہے اور یہ ہمیں شو کر رہی ہے سائنرجی. اور اس کے علاوہ تھرڈ اسٹین جو ہے MSSA اور یہ ہمارے ساتھ ریزلٹ شو کر رہا ہے ایڈیٹیو. سو یہ ڈیٹرمنیشن آف فوڈ یہ ہے مختلف. یہ فرسٹ آف آل ہم نے اسٹین کی وہ بھی معلوم کی ہے اور اس میں CFU جو ہے نیچے CFU ہے 0.09 میں CFU 1344 ہے اور 0.06 میں 1196 ہے اور 0.04 میں 910 ہے. اور اس کے علاوہ جو ہے اینڈ سو آنس یہ ہمارے ریزلٹ سے 0.09 کے CFU اتنے اس میں اتنے قانونی کاؤنٹ کیے ہم نے اور 0.06 میں ہم نے اتنے مثال 1196 قانونی کاؤنٹ کے 0.04 میں 910 اور 0.03 میں 808 اور 0.02 میں 603 قانونی کاؤنٹ کے 0.01 میں 43 مثال ٹائم کیلے سے. تو فرسٹ آف آل ہم نے جو کاؤنٹ کیے ہیں یہ ڈیکریز ہوتے جا رہے ہیں، ڈیکریز ان کلنگ ہو رہی ہے اس میں. تو ہم یہ شو کر رہے ہیں کہ جب کلنگ ہوتی تو ہمارا ریزلٹ گڈ ہے. اور یہ ہم نے 100 فولڈس کیے ہیں، 100 مائکرو لیٹر سیریل ڈائلوشنس کیے ہیں اور یہ بھی ہمیں سائنرجزم ریزلٹ دے رہے ہیں. یہ کمبینیشنز ہے 2.12 کے اتنی CFU ہے 0.09 کے اتنی CFU ہے اور 0.06 کی 1167 اتنی سیچی جو آپ 0.03 کی 787.5 ہے اور 0.01534.5 یہ اس کی CFU ہے. اور جو فرسٹ آف آل ہے 0.12 ہے 0.09 ہے 0.06 ہے 0.03 ہے 0.01 تو یہ بھی ڈیکریز ہو رہا ہے. CFU جو ہے 1926 اور 1546 1167 اور 787 اور 534 یہ CFU آئے تو یہ بھی ڈیکریز ہو. جب ڈیکریز ہو رہا ہے تو اس کا مطلب یہ کہ یہ کلنگ کر رہے ہیں تو 100 فولڈ سے تو یہ بھی ہمیں سائنرجزم شو کر رہا ہے. سو مائی کنکلوژن یہ ہے کہ فیوچر مثال synergism activity of gentamicin like lavender and have shown the broad spectrum against various microbial agents like bacteria, fungi, and virus. So future study will be for a good effect of gentamicin, lavender oil, and also used for cosmetic purpose and with minimum or no side effect. So this one, my presentation shortly, and thank you for listening my presentation. Thank you.
Thank you, Habibullah, for your nice presentation. Our next presentation is by Nimra Zafar with title Histological Effects of Lactuca cereula on Vaccine-Stimulated Immunity Against Newcastle Disease Virus in Chicken. Please, Nimra Zafar.
With the name of Allah, the most merciful and beneficial, I am Nimra Zafar from Government College, University of Faisalabad, Department of Zoology. Here I'm going to represent the topic of my presentation is histopathological effect of Lactuca cereula on vaccine-stimulated immunity against Newcastle disease virus in chicken. Poultry industry is the second largest developed industry in Pakistan and one of the affordable and considerable source of protein as egg and meat. All around the world, poultry is facing many socio-economic losses due to viral poultry disease, with every passing year like Newcastle disease is one of them. It is commonly in Pakistan, causing potential economic losses throughout the year. What is Newcastle disease? Newcastle disease is highly contagious viral disease of avian species and represent a major threat to poultry industry worldwide. It is caused by RNA virus, which is classified in genus Ebola virus family Paramyxoviridae. In Pakistan and India, it is commonly known as Ranikhet. About 250 bird species of 27 order are affected from this virus. Morbidity up to 100%, mortality 90%. The clinical signs and symptoms are as shown in this picture: edema of head, especially around the eyes, edema around the neck, edema around the head, hemorrhage lesions, green watery diarrhea, drop in egg production. Surviving bird may have neurological and respiratory damage and sudden death. Alternative to chemical drugs, Pakistan is still spending huge revenue for importing ND vaccine, which is more costly and can't affordable by local farmers. Currently, control of NDV is normally achieved by vaccination program with inactivated and live attenuated virus, but outbreaks are constantly arising in spite of costly vaccination. The basic need of the time is to discover new corresponding actions and antiviral agents that not only effective and safe but also overcome resistance and cause toxicity. Scientific investigation to invent new antiviral natural products which can protect humans and animals against infectious disease. According to World Health Organization, 75% of the world population are using herbs for the basic health necessities. Pakistan export high-value plant generate over 10.5 million annually. Europe, as this annually import about 1 billion. Percentage use of the medicinal plant widely used in human therapy, by veterinary, agriculture, scientific research, and countless other areas. This is the valley which I have collected Lactuca cereula parts of the plant. When Soonskesar Valley is the part of the salt range, Soonskesar Valley has a unique kind biodiversity involving distinctive climatic zone and wide scope of the plant species. The inhabitant of the Soonskesar Valley has a rich convention of restorative plant utilization in this unique situation. 15 plant species of the 10 families investigating which are utilizing by inhabitant of Soonskesar Valley for their regular day illness and furthermore to get numerous other value included product. Commonly known as the wild lettuce, kahu and khas, it is family Astraceae and native to Himalaya, Siberia, Atlantic areas but cultivated also in temperate lands of Europe, India, Pakistan and Iran. The objective of my study is to evaluate in vivo anti-NDV activity against causal disease virus and to study histopathological changes in trachea, lungs, and cecal tonsil of the chicken. Workflow plan: first of all, after being collecting clean, hygienic, and disease-free part of the Lactuca cereula from Soonskesar Valley, the plant used dried and ground in a grinder for being dried. Powder plants were carefully dried in the shade and then methanol and chloroform used in the Sockley extraction. To extract the plant powder dried from fine powder placed in a thimble extractor, and then after extract the process, after completing this process, extract was viscous using rotatory vaporizer and then prepared concentrated extract. This one is the experimental layout. A day-old unvaccinated chicks were divided in 7 groups of 5 chicks in each group and then acclimatized for 1 week with complete water and feed was offered. At 8th day, group 3, 4 and 5. Group 3, 4 and 5 were vaccinated with Lasuta strain of NDV with eye drop 50 microliter. Then after 2 days, group 4 and 6 vaccinated and treated with the 100 mg per kg of body weight Lactuca cereula methanolic extract in drinking water. After group 5 and 7 were treated with 50 mg per kg body weight of Lactuca cereula methanolic extract for 5 successive days. At 16 days, the challenge strain of NDV titer was given to all group except negative control group. And the general observation is to that starting from the first day of the administration of the plant extract, the chicks were observed well for their general performance in order to evaluate in vivo viral activity measurement. In vivo viral activity like FCR, body weight, histopathological measurement were evaluated from the survived chicks. After that, after treating with the NDV challenge strain, then the clinical sign and symptom were observed. The clinical sign and symptom were observed as you can see in these pictures. All the chicks are healthy till 11th day. On one day after NDV strain, various signs of viral infections shown included torticollis, as you can see in this picture, white watery fluid, from mouth, depression, wings and legs paralyzed, hemorrhages on foot, and death, sudden death. So 80% of the chicks' death in the positive control group, and 50% of the chicks' death in the vaccinated control group, and 20% in the chicks' death in both treated challenge group and non-vaccinated. But in negative control group and vaccinated treated challenge group with plant extract has showed 0% mortalities. Effect of Lactobacillus cereula on body weight and food conversion ratio. Effect of the Lactobacillus cereula on body weight has been result that indicate that Lactobacillus cereula significantly improve the performance of the treated group, as compared to the other group as shown in the table. And food conversion ratio, it was revealed that there was no significant difference in feed intake of treated challenge group, group and control group, so suggesting that the Lactobacillus cereula plant has no effect of food consumption ratio, but in positive control significantly worse ratio in the in this table as compared to the other group. Macroscopic lesion of the trachea, in which macroscopic lesion of the trachea after slaughter the chicks, different abnormalities examined in the chicks part of the trachea, as like tracheitis, bronchitis, necrosis, and exfoliation of the mucosal epithelium, fibrous exudates, and cellular debris. No obvious histopathological lesion are detected in the negative control group, but in the positive control groups where lesion histopathological alteration are caused in the positive control group, as you can see in this table. In treatment challenge group characterized by the different site, as you can see in this picture, like A is a negative control group has no obvious changes, but in B severe hemorrhages in tracheal mucosal lumen with the sludge mucosal cell necrosis and infiltration of the lymphocyte. In this pictures, it is a vaccinated control group. Arrow indicate extensive dropout, degeneration, and necrosis of the ciliated epithelial cell. And this one is showed deciliation and hyperplasia of tracheal mucosal epithelium and congestion, and both are the vaccinated treated challenge group. And these are the plant extract, give the plant extract, and extensive deciliation and lymphocyte infiltration with the moderate congestion. And this one is treated challenge group, mild deciliation of the tracheal mucosa and mild inflammation reaction. And this one is the treated challenge, but 50 mg per kg hyperplasia of the tracheal mucosa with severe edema and congestion and hemorrhage. Next one is macroscopic score of the lungs. Macroscopic lungs showed moderate to severe lesion in different groups. As you can see in this picture, 2 and 3 positive control and vaccinated control is moderate to severe, and group 6 and 7 are treated challenge group, 100 mg and 50 mg mild to severe lesion are caused. When vaccinated treated challenge 150 mg showed less lesions in this group. Moderate lesion recorded in the vaccinated control group, moderate lesion, and the higher and severity caused in the positive control group. And total treatment challenge group shows rupturing and collapsing of the alveoli with the congestion, as you can see in the picture, in this picture it is a negative control group. It is a normal section with no any inflammation, but this one is the positive control group, severe hemorrhages and necrosis of the epithelial lining, the parabronchus and edema, which shown in this positive control group. And this one is vaccinated group showed sludging and epithelial lining the parabronchus with the congestion of the blood capillaries. This one is the vaccinated treatment challenge group showing mild congestion of blood capillaries, and this one is vaccinated treatment challenge with 50 mg per kg with mild edema, and both are the treated challenge group 100 and 50 mg according to this ratio, and these are showing this one are showing rupturing of the alveoli and mild hemorrhages, and this one is showing mild congestion, collapse, and rupture of the alveoli. Next one is the macroscopic Lyon score of the sickle tonsils. Macroscopic score of the sickle tonsils, as you can see in this picture, more severe Lyon shows in the positive control group as compared to the negative control group, and less severe in the vaccinated treated challenge 150 mg per kg from rather than the treated challenge group. As you can see in this picture, this one is the negative control group and showing the normal cell, and this one is the positive control group, severe necrosis of epithelial lining and lymphocyte, fibronohemorrhages, absence of hemocytes, and this one is the vaccinated group showing extensive hyperplasia of the lymphoid follicles. And then both are the vaccinated treatment challenges with 100 mg and 50 mg per kg according to this ratio. This one showing mild depletion of lymphoid element, and this one showing depletion of lymphoid follicle with lymphoblastic activation, and both are the treated treatment challenge treated with challenge group 100 and 50 mg per kg according to this ratio, showing severe lymphocyte and necrosis of majority lymphoid element comprising the follicle, and this one is showing the congestion. At the end, conclusion is that NDV is a serious threat to the global poultry industry due to high mortality rate. Reproductive, respiratory, neurological, and gastrointestinal indication are the most common clinical sign of the ND. Most of the medicinal plant has been used for the treatment of the bacterial, fungal, and viral disease among the different animal, including human. Methanolic extract of the Lactuca cereula in chicken was applied against NDV as antiviral treatment, and found that this plant has no harmful effect on growth performance, body weight, FCR, and reduce the histopathological damage in the chicken. So conclusion is that using methanolic extract of Lactuca cereula for NDV in poultry has the potential to significantly reduce the clinical and histopathological aspect associated with the disease, hence decreasing the economic loss associated with it. Thank you.
Thank you, Nimra Zafar from Government College University Faisalabad for your nice talk. Our last talk of the session 7th is by Muzammil Hussain from Government College University Faisalabad with title Exploring the Rheological Properties of Arabinoxylans Isolated from Different Cultivators of Psyllium Husk. Please, Mr. Muzammil Hussain.
Assalamu alaikum. Welcome to 4th International Conference on Applied Zoology 2021. I am Muzammil Hussain, PhD scholar, Government College University, Faisalabad. Today, I will discuss about presentation in 4th International Conference on Applied Zoology, title Exploring the Rheological Properties of Arabinoxylans Isolated from Different Cultivars of Psyllium Husk. And today my agenda is firstly I will discuss about the production section of Psyllium Husk, and then objectives and materials and methods used in whole work, and then describes results and discussion section, and finally at the end conclusion section must include Plantago vata Psyllium Husk. locally known as Ispavol, scientifically known as Plantago ovata. It is a medicinal plant, common source of psyllium seed or psyllium husk. It is a total dietary fiber pest herb that is used to treat stomach-related diseases. Psyllium husk derived from the seeds of the Plantago ovata. It is a fiber supplement that reduces constipation due to its excellent water solubility. Help to regulate high cholesterol, triglycerides, and blood sugar level. It also helps weight management and relieved mild diarrhea. Here is arabinoxylin. It is a totally hemicellulose, mainly present in cell walls of different plants and cereals and husk. Psyllium husk contains dietary fibers and arabinoxylin. It is a totally psyllium husk is a totally dietary fiber and arabinoxylin. Psyllium husk is the principal source of arabinoxylin. As a dietary fiber having many health benefits, including antioxidant, anti-cancer, anti-inflammatory, antimicrobial, anti-aging, and cardioprotective, prebiotics, and antiviral effects. It has many functional and nutraceutical components like arabinoxylin and functional different phenolic compounds, ferulic acid and synergic acid, and also having many functional and rheological properties. And here is my objectives. To explore the nutritional composition of psyllium husk and to isolate arabinoxylin from psyllium husk and to prepare arabinoxylin in rich flour and also explore their rheological properties. Here is my research design. Phase 1 explore the biochemical or nutritional profile of psyllium husk and then extraction of arabinoxylin from psyllium husk and then addition of arabinoxylin in flour and finally explore the rheological properties of. Here is material methodology section. Two different varieties, Pakistani and Indian psyllium husk procured from local market of Faisalabad. Chemicals procured from the local market of Faisalabad. 100A was procured from Ireland. And then here is the chemical analysis of psyllium husk. A biochemical analysis of psyllium husk from different varieties were analyzed for approximate analysis, especially moisture content, protein, fat, fiber, and the total dietary fiber, soluble dietary fiber, and insoluble dietary fiber were determined. And extraction of arabinoxylin. Here is the method of enzymatic method of extraction method. Ispavol husk. In first step, take ispavol sample and then heat treatment 130 temperature for 19 minutes by using hot air oven, and then remove of solubles by using centrifuge at the conditions of 3200 G and for 10 minutes, and then the enzymatic treatments was performed by using PBS buffer solution and by using and also used different enzyme used in this process is protease enzyme and amylase enzymes and also use glucosidase enzyme, and then washing and remove starches, proteins and sugars, and finally extract arabinoxylin by using centrifuge. Here is the treatment plan. T0, T1, T2, T3 and T4 Here is the treatment plan based on the arabinoxylin addition in flow. T0 100% floor used, and T1 5% arabinoxylin and 95% wheat flour, and T2 10% arabinoxylin and 90% wheat flour, T3 5% arabinoxylin Indian varieties and 95% wheat flour, T4 10% arabinoxylin Indian varieties and 90% wheat flour. Rheological properties, phrenograph and mixographic studies, dose properties of different wheat flour blend containing arabinoxylin in different concentration was determined by using phrenograph. And mixography graphic studies was performed by using a mixograph. And statistical designs, the data of each parameter was subjected to statistical design to determine the significance, least significance, and one factorial analysis was performed. Here is the results of my research. Biochemical compositions of different two varieties, Pakistan and Indian varieties of Symptom husk. Here is the Pakistani varieties has higher moisture content 4.45% and higher ash content present in Indian variety 3.9, and crude protein present in Indian varieties 3.86. and fat is 1.42 present in Pakistani varieties, and fiber content in Indian varieties is higher than Pakistani varieties 17.20%, and NF is 63.4% content in Pakistani varieties. And the total dietary fibers is higher in Indian varieties 52.5%. Then 49.5% Pakistani varieties, and soluble dietary fiber content higher in Indian variety 3.48, and insoluble dietary fiber content in Indian varieties is higher 49.62% by following Pakistani varieties. And here is arabinoxylin content. Pakistani varieties 34.52%, but Indian varieties has higher arabinoxylin content 38.92%. Here is the phrenographic results. Phrenograph water absorption. Here is water absorption capacity. 60 treatment plant T0 100% wheat flour 62.38% water absorption, but after the addition of 5% arabinoxylin Pakistani variety 65.2%, and water absorption capacity increased by adding arabinoxylin, and after 10% addition, and the water absorption is 69.61%. And then the same trends present in Indian variety 5%, 66 and 34, and T4 671 to 83%. Here is the water absorption increased due to arabinoxylin, because previous studies or previous research showed that arabinoxylin has higher water absorption capacity than wheat flour and other fibers. It has more than 80 to 11% water absorbed to actual weight and the dough development time was decreased after the addition of arabinoxylin, dough stability time, and the MTI and S results. There is mixographic results. The mixing time is important in industrial scale. Here is the table showed that results showed mixing time of T control is 66.31, and then the T1 time reduced 6.02, and after 10% addition 5.83, and then Indian varieties showed the less time 5.22%. Conclusions, psyllium husk is an excellent source of dietary fiber, especially arabinoxylans and other beneficial complexes. Arabinoxylan of psyllium husk tend to have positive effect on rheological properties of flour, farino, and mixograph. Especially rheological properties, psyllium husk, arabinoxylin increase the water absorption capacity. It is beneficial for industrial scale because it has absorbed more water, and also effect on the final product quality, like different baking products like bread. Recommendations. Psyllium husk hold potential for helping the future generation to meet their nutritional needs. Psyllium husk arabinoxylin contains several beneficial components like dietary fibers, arabinoxylin, that should be added as a functional food, as a in many functional food products tend to prevent and cure various chronic amendment, and consumer are learning more and more word to the natural remedies. Arabinoxylin should be added to the bakery goods for the production of functional food. Thanks for your attention.
Thank you, Mr. Muzammil Hussain, for your nice talk. Keeping the time in mind, our 7th session of ICAZ 2021 is going to its end. I am thankful to all the researchers, scholars, faculty members, and viewers for your participation and attentions. With the end of our 7th session, all the activities, programs, and sessions of Day 1 of the 4th International Conference on Applied Zoology 2021 will also complete. I hope first day of the ICAZ 2021 was productive and you got a lot of information. Our next and last day of the conference will start at 7 a.m. at Pakistan Standard Times. Till that, stay happy, stay tuned. See you tomorrow at sharp 7 a.m. Allah Hafiz and goodbye.